Good morning.
Whoop.
I
All
can.
right. It doesn't like the video, but that's okay. So I just wanted to welcome everyone to the Winter Weaves meeting. This is being recorded, so I've had a few requests for that, and I'm happy to say that we can do that. For those who've been in the organization for a few years, you know that traditionally this was an in-person meeting held up at North Bend State Park. And then in the past few years with COVID, we decided to do it virtually. And last year it was a really big success. We had a lot of folks that were able to join us virtually that maybe couldn't have joined us in person. So we decided to do that again this year, which I'm very, very happy about, given the amount of snow that we got all over the state and then the continued issues with COVID. So I would like to just welcome everyone joining us. And I tried to put together a really interesting list of speakers, and I really got a few folks who we've never seen before in this organization. So I wanted to get a little bit of new blood in, and I encourage you all to talk, in the chat with each other. I'd ask that you mute while someone is presenting. I've built in plenty of time to the schedule for questions after. So if you could hold your questions until the end. When the speaker's done, I will open it up and we'll take questions for a few minutes. So we've got about a half an hour a speaker. So I think we'll be okay on time. Karen, am I forgetting any major announcements before we start?
No, I don't think so. Sounds good.
Okay. Well, then I would like to present our first speaker of the day. This is Kristen Wickert. Some of you may know her from her former position as the Spotted Lanternfly Coordinator for the State Department of Agriculture in West Virginia, but she is now taking a new position and is dealing with pollinators for Appalachian headwaters. And the title of her presentation today is going to be Arthropods and their relationship with fungus. So take it away, Kristen.
Thank you. So I am going to share this and I'll double check with everybody that it is moving and grooving once we go. But yeah, Rachel did a great job describing who I am and who I was, and I hope to always be involved in the world of spotted lanternfly as long as it exists anyway. But I have worked for a really long time with a bunch of different arthropods, and I know you guys are all insect experts, so that's why I didn't call this insects in their relationship with fungi. We're going to talk about all of our jointed leg friends. And in order to do that properly, we have to talk about their different relationships, which are deemed by myself the good, the bad, and the ugly. And in my presentation, the good will be these dependent and symbiotic relationships. The parasitic relationships are, of course, not very good for our arthropod friends. And then the ugly will be the vector job, because sometimes the vectors can look, ugly for us, especially in the world of plant pathology and invasive species. So we're going to start with the crowd pleasers, because everybody probably sees this and they go, ooh, how exciting. And they are very exciting. These are the citrus cicadas that have been infected by a fungal pathogen. And these are the nymphs that would normally be crawling around under the ground. You can see they're covered in some debris and dirt. And they would be under the ground, but the fungus emerges above the ground in order to spread its spores. And when we cut one of these cicadas open, you can see that their entire insides have been utilized and replaced by their fungal assaultant. And what we call this is an entomopathogen. And an entomopathogen is the combination of the two words ento and pathogen, which means insect disease. And what's happening in most of these scenarios is that a fungus will exist as a spore because they reproduce by spores, and they will land on an insect's body and penetrate through their hard chitin shell or their exoskeleton. They'll penetrate through there, and then they will populate and proliferate inside of the insect's body, from being a yeasty mess all the way into turning into fungal hyphae. And once they've eaten the whole inside of the body, like what we saw with those cicadas, the fungus will emerge out of all of their joints and everything, any weak point in their exoskeleton, and produce spores and start the cycle all over again. And a couple of cool North American examples that we have in West Virginia. By the way, most of these photos have been mine so far, and they were all taken like this year. So you can find these outside. This is the very popular Cordyceps militaris. The orange fruiting body here is releasing spores, and it emerges out of a lepidopteran, probably a noctuid moth pupa. Another really common example at the end of summer are these aggregating flies that are all like covered in the fruiting spores of Entomophora muscae. And if any of you are familiar with Latin, muscae is the word for fly. So it's in a pathogen of flies. This is super common right now outside. If you go for a walk, it's buried under the snow right now, but during the winter and the fall, and you can even see this background here is a red leaf that I used as a background in the fall time. This is Acanthomyces accal, which totally fills up the body of the moth as an adult, and it will bust out of all of those openings and joints, and also along the main veins of the wings. When you look really closely, and I know a couple of you work for the Forest Service and Department of Ag, if you look for Hemlock woolly adelgid, you might get confused and see this little fluffy white mess. Well, this is actually a spider that has been infected by the Gibbelia fungus, and it exists in two different forms and how it spreads its spores. So depending on if you are looking for HWA in the summer, you'll see it in this form, and then in the winter, you'll see it in this form. And a really common one that is a super generalist in West Virginia and most of the United States and world is Beauveria bassiana. And Beauveria bassiana can be on multiple hosts. So here it is on crane flies. This was a picture taken at Cooper's Rock. And there were many crane flies all in one spot on a log that were all infected with Beauveria bassiana. And as a generalist, it infects many different hosts. And it actually has been found killing spotted lanternfly. These were some spotted lanternfly that were found in Philadelphia that were sent to me. And the fungus is puffing out of their body, producing spores, which if you look really closely, I took a dissecting scope and put them under there. And you can see all of the individual little spore masses. And here they are on a slide. So this has the potential to be used as a biocontrol for an invasive pest species. And during my master's program at WVU, I looked at Hemlock woolly adelgid and tried to find fungi that might be utilized as an entomopathogen against Hemlock woolly adelgid. And I discovered a couple of facultative entomopathogens that have the ability to live in hemlock tissues, but if they are in the correct environment or close to an insect, they possibly can infect an insect, which is really interesting because it means that they have the genetic ability and the enzymatic activities to break down insect chitin, which fungi are also made out of, and to infect and also utilize a different food source than cellulose, what they're normally enjoying in plant tissues. There's a really popular example of entomopathogens in the news. You'll commonly see this, or like just clickbait on the internet. Summit disease is something that fungi can do when they infect insects, and it is the famous mind control of the Cordyceps fungus. And I have quotations here because Cordyceps fungus, Cordyceps is a genus that is a popularly known genus. However, a lot of these examples are not Cordyceps, but they just get lumped into there. But the summit disease is when a fungus causes an infected insect to climb high, it's summiting, to high locations before it creates spores or a sporophore structure here, because the wind will help the spores blow throughout the environment and infect more insects. And sometimes it can even be as specific as making the insect crawl back into its nest or its hive to try to infect other members of its hive, like zombie ants. And we are learning more and more examples of summit disease all the time. And unfortunately, they focus a lot on the tropics, like in Cambodia and in Costa Rica, but we have examples of this all over the world. And that is the first example of what I got interested in or involved in when I was young and able to climb mountains all day. I used to survey for a Lepidopteran larva, the Aroeus occulta larva, which was a caterpillar that performed summit disease when it was infected with a fungus. And there aren't many trees at all in Greenland where this picture was taken. And so they aren't summiting on top of a tree or a really big cliff, they're summiting on top of blades of grass. And then the fungus will come out of their head and spread to other caterpillars in the area of the same species. And I continued to be involved with the entomopathogen story and discoveries throughout my time in graduate school. And we discovered throughout that time, it's been discovered that many different orders and genera of insects are infected by many different orders and phylum members of fungi. And one of those really exciting stories was the recent discovery of the genus Massospora having chemical abilities to influence the periodical cicadas, which happened last year in West Virginia. And this fungus, Massospora, affects both periodical and annual cicadas in the United States, different species in that genus Massospora. But what this fungus does, it affects the sexual reproductive organs of cicadas. So in this bottom picture here on the right, there is a female cicada with her ovipositor, and then there is a male cicada over here where his reproductive organs have been completely consumed by the fungal tissue. And it has a unique behavior modification. It's kind of like a summit disease, but it doesn't, it's increasing the way that the fungus spreads, but it doesn't like kill it and make it climb up. It actually makes male cicadas act like female cicadas by clicking their wings and no longer singing. Only the male periodical cicadas sing, but the females will respond by clicking their wings, and the fungus mind controls them to make them act the different gender to attract other mates, so that way they can spread this STD fungal tissue here. And all of this is happening while the cicada is still alive. And it's not uncommon for at the end of summer for you to see a periodical cicada walking around without any of its abdomen anymore because it's been totally utilized by the fungus. So let's move on to the next relationship, which I deem as, the good. And that is probably the most obvious, even though it's the most ignored because it's not exciting in the media. But it's really obvious that insects and arthropods utilize fungi as food. And this is a really interesting example for me as being a bug and mushroom nerd, where there's an entire family of tiny little coleopterans that specifically inhabit fungal polypores. And this example, and this is from West Virginia, this was in Morgantown, this is my old apartment here, where there was a cracked cap polypore, Pholinus robiniae, which grows only on black locust. And inside of it were all of these little larvae of the beetle that were burrowing around, creating frass. And also, there were adult beetles in there living all throughout this polypore. So they utilize it as food. And it's very hard to find information about this group of beetles, but this is the only observation of this very small, less than only a millimeter wide beetle on iNaturalist, which I really suggest you guys start using. A very big common example with a lot more observations are the pleasing fungus beetles. Again, they're coleopterans, and they feed directly on polypores. So this is a big mushroom that this beetle is resting on. But mostly tiny things directly rely on fungi for food. So it requires you to sit down on the ground and take a couple of minutes to look at things. This is a twig that had the amber jelly roll fungus on it. This is, its scientific name is Exidia recissa, and it's covered by little springtails that like to feed on it during the winter when there's not very many other things out. But we can get even tinier, and this is an example of why this talk is called arthropods and their relationship with fungi and not insects. Because if we look at this Petri plate that was in my lab, I must have gone on vacation and not paid attention to a plate for a while, but all of these little lines, these little dots, are caused by a very small mite. So these little blobs here are actually something called a fungus mite, which solely relies on eating fungal cultures. And if you get them in the lab, you need to just throw out everything and can decontaminate everything because they spread like crazy. So this direct dependence can actually lead to a symbiosis where fungi and the insect both benefit instead of just being something that the organism eats, right? And this is a beautiful millipede, again, arthropod, not insect, but this is a beautiful millipede in the genus Brachiocybe. This particular photo is when I lived in California. This is the Brachiocybe rosea, and it is feeding together as a family unit on, with its little babies here, on fungi that it actually cultures and maintains and farms. And the lab I was in before used to work on these Brachiocybe millipedes, and it was a really fun time, but there was also a lot of work down at Virginia Tech and University of Florida that studies these Brachiocybe lecontii that are on the East Coast. And it's been, we've been studying to see if there are different reasons that these fungal farming millipedes actually have many different fungi that they take care of. So in this picture, there's actually 3 species of fungi. There's this bluish one here, this purpley gray white one here, and then underneath them is a black one. And it's hypothesized that depending on how the health of the colony, they might feed on one fungus that has nematicidal properties or another fungus that they just get carbohydrates from. So it's really interesting that they both rely on each other to take care of each other and continually propagate their species. And these arthropod fungal symbioses can become incredibly entrenched and co-evolved. I know a lot of you are very familiar with the tiny beetles of the Curculonidae family. And you've probably seen this picture from my old advisor back at WVU. This is a slice, a microtome slice of the head of a Eualacia validus beetle that actually shows their like cute little chipmunk cheeks, which are called mycangia on this species, where they actually collect fungal spores and purposely inoculate trees so that way the fungi can eat the food for them, which is a tree. And because the beetles don't have the ability to break down cellulose and lignin in their body, but fungi do. And there's a lot of these small little bark beetles and ambrosia beetles that have different types of mycangia. So instead of being in their cheeks, this species here has these 2 little holes on its back, where if you look through a dissecting scope, these little pockets are filled with fungal spores. And so when that beetle is burrowing into the woods, it's scraping its back onto the exposed wood and infecting the tree that its babies will then eat the fungus. And this is another study from right outside the door of WVU, right outside of our old lab's door, where there was a tree of heaven that had some beetle galleries in it. And this is what I'm talking about. The fungus is able to eat and digest the wood, and then it creates a spory mass here for the beetles and their larvae to crawl around in here and directly eat the fungus. So they're animals, they're just like us. We can't eat wood and digest it, but we can eat mushrooms. So that's just an easy, fun example of a super symbiotic relationship. And sometimes these relationships can get so intense and co-evolved that no one can leave. And this is not my work, but it's just a really interesting example where the leafcutter ants that are in the tropics, but also in like Texas and southern parts of the Southeast, the leafcutter ants will cut out leaf parts and bring it back to their fungal comb inside their fungal garden inside of their hive, which is underground. And the ants no longer have any enzymes that they can directly eat leaves themselves at all. And the fungus doesn't even reproduce spores anymore because it doesn't need to reproduce. It physically just gets taken care of and moved around by the ants into new colonies. So without the ant, the fungus would die because it physically can't reproduce anymore. And then the ants couldn't live without the fungus because they can't eat anything else. So the final example in this talk is the job of being a vector. And sometimes that is ugly to us. These insects and fungi are just doing their own thing, right? But because we have changed the world and made it into a large monocrop cereal world, we don't like it when insects carry fungal pathogens around. And so here's an illustration from this paper of beetles as plant pathogen vectors, where a beetle will chew on a leaf because it's eating, but almost unknown to itself, inside of its gut are a bunch of bacteria, fungi, nematodes that might act as symbionts inside of it to help it digest, but also might just be relying on this winged creature to take it to a new plant, and then it gets into the plant that it eats instead of interacting with the beetle at all. But just a fun example of how fungi can attract these insects to make them spread them around. These are 2 species of stinkhorn that are in West Virginia. There's a couple of more species, but these are the most common ones you'll see. This Phallus rubicundus was actually taken at Cooper's Rock, and I took this on another hiking trail close by, but they both grow in like woody mulch. You might see them actually outside of buildings sometimes if they have fresh mulch. But if you look at it, you can see all of these flies on top of this Phallus duplicatus, and they are spreading these spores because they're attracted to a stinky smell that'll make you gag. The flies will step on the spores, fly away, and land on more mulch. And then on this, it's kind of harder to see, but there's 2 little fruit flies on top of this different stinkhorn, which has a different stink profile, still nasty to humans, but flies love it. And sometimes we can use this ability of fungi utilizing arthropods as vectors to our advantage. It's also scary because they spread diseases, but sometimes we can use them as an ally. So this is an example of my PhD work, where this is a site that is completely overtaken by an invasive tree species called tree of heaven. And I utilize a fungus that was a biocontrol for this invasive tree species that would kill the tree. But while it was killing the tree over the course of a couple of months, the tree would exude a lot of ethanols and other kinds of smells that would attract a exotic naturalized coleopteran species, Eualacia validus. And these beetles would be attracted to infected trees that were sick, that had the fungus in it, and the fungus would get on their bodies, and then they would fly to nearby tree of heaven and infect it. And you could quickly see the spread of this biocontrol when these beetles showed up about 2 years after the tree of heaven started to be really stressed out. So that would lead to sites like this, where perhaps I actually only utilize the fungus on this tree here to kill it, but then I would see many other trees with some other reasonings for that happening, but there would be a lot of beetle holes in these trees because they make those little frass noodles. But with that, that's the end of my talk. It looks like I have about, it's about 10:23, so I have a couple minutes for questions. So go ahead. Thank you for your attention.
Thank you so much, Kristen. That's fascinating to hear about all the different relationships.
Oh, I love them.
Do we have any questions? You can either put the question in the chat or just unmute for a second and ask Kristen what you need to ask.
On one of the images of those millipedes feeding on fungus, I noticed an odd little mite. Do you know what kind of mite that was?
No, but I was wondering who would notice that. I'm so glad it was you, Ed. It's nice to see you.
Yeah, that was a great talk, by the way. Really enjoyed it.
Oh, thank you. I have an inkling to guess that I think there's actually something called a long-legged mite. I think that's what it was, but I've not looked into it. I do not have a mite key. So, but I have seen something like that before, just when I was cruising around the internet.
Okay.
So there's a question in the chat from Michael. It says, "Hi, Michael, nice shirt." It says, "Thank you for the presentation. I appreciate that. Thank you. Does the pathogen for Tree of Heaven also affect other trees?" My PhD work was a continuation of like 4 other people's PhD works. And over the course of that, we really expanded on each other's research. And there were 2 huge host range studies that included over like 400 tree species, both native to Eastern North America and then exotics, because it also wanted to include like trees that people had in their front yards and then like, ornamental stuff at botanical gardens. And there were a few species, and by few, I mean like 3, that experienced symptoms. However, they recovered, and the symptoms were minor. So the trees fully recovered because we were able to go back with we all knew each other throughout our PhDs, and I even worked for them as a young person. So we were able to see that it didn't infect or it didn't kill other tree species. And then I also did a lot of genetic work to prove that there were specific proteins associated with how they kill Tree of Heaven, how the fungus kills Tree of Heaven.
Follow up to that, the beetle that would spread that fungus, would those attack other species of trees?
Yes, they do. And that's always been a really interesting thing for me now that I don't have a lab anymore. I really wish I did. I have so many questions all the time when I go out in the field, but actually last year, or when I was looking for spotted lanternfly at the I-81 rest stop on the southern one, I actually found a honey locust that was right next to some Tree of Heaven that had Eualacia validus holes in it, but and it's because the beetles, they really can't see that well, right? They can smell, so they head towards the smell, and then they just, I think whatever they hit, they're like, oh, this is the one. And so right next to some of the Tree of Heaven, they had gone into a honey locust, and there was a lot of pitch that was coming out of the honey locust hole and pushing them out. And I don't know what will happen with that. And that kind of builds off of, like, you're right, Michael's question. Even though we did direct inoculations with like really high levels, maybe there's some kind of interaction that could happen from those beetles spreading it to something else. It is possible, because Eualacia validus, which is that really small little beetle that spreads the disease, it's not using the fungus I used as a biocontrol in its own existence as an ambrosia beetle. It utilizes a fusarium. And so that might be something that happens where the beetle is attracted to the Tree of Heaven that's dying. It has my fungus over its body, but it also has the fusarium in its pouches. And then when it gets confused, maybe it'll go land on that honey locust, and the fusarium is actually what kills the honey locust.
Okay.
If someone were to give me money and time, if someone were able to give me like 9 more hours every day, I would do it.
Hello, Kristen. This is Eric from
Hi, Eric.
DHHR. Hey, I was going to ask, the fungal infection associated with the magic cicada. Did that have any negative impact on any vertebrate host by any chance? Do you know?
Negative impact on any invertebrate host?
Vertebrate, like birds is what I'm thinking.
Oh, no. And also that has not been completely exhaustively studied yet, because this discovery did happen relatively recently.
Yeah.
So it was like 2016 when the cicadas were actually in Morgantown when we were all in school. And that's when we started to make the first observations. And then with the brood X or brood 10 happening in 2020, which is the biggest brood, that's when it got a lot of attention on the media, because brood 10 was like all over the whole East Coast. Whereas when we discovered it in 2016 to 2017, our brood was really small and it wasn't in the media. But it hasn't been noticed to infect any birds or anything, but I also don't think anyone's really set up a trial, because you have to like think about how far a graduate student would have to drive to follow broods, right?
This happened a long time ago. This had to deal with what we thought was early bird mortality. which coincided with the magic cicada emergence. And there were crazy ideas that perhaps maybe the birds are eating these magic cicadas, which may be infected with the fungus or pesticides. And
Well, go ahead. Sorry.
oh, sorry about that. DNR really doesn't think that's what happened, but
Mm-mm.
and it's really not looking that way. And they were even thinking it could have been due to West Nile, and that was found to be incorrect. So we're just
Well, if you
curious.
think about it, again, disproportionate stories from the media, you would think that these cicadas are all infected, but actually it's only like 1 to 3% of the magic cicadas get massospora infections. And it's happening at the end of their adult life cycle. So it's like, I really didn't see that many this year. And I was out in the field every day looking for spotted lanternfly where the cicadas were. And then another thing is that most of the birds and other things are already like, they're like, forget the cicadas. Like, I'm tired of them by the time that the massospora is showing up.
Thank you, appreciate that.
Yeah, thank you.
Okay, any last questions for Kristen?
One last thing, and in going through caves, I've often encountered the cave spider, Meta ovalis, covered with a white fungus. Would that be anything different from what you'd find outside on the surface?
It's probably a very adapted species of Gibellula. Remember the picture I had with the 2 spiders on the
Yes.
hemlock? So Gibellula affects many different species of insects, but it has a preference for spiders. And that would be my guess. I have seen that. Have you ever been to the Sphinx of Gandhi?
Yes, many, yes.
Yeah, so in the Sphinx of Gandhi, the entire ceiling was covered in those fungally engulfed spiders. And my guess would be that it would be a Gibellula or it would be the Beauveria, but it's probably a super specific species that can live in caves and not have any UV interactions, you know what I mean?
Okay. Thank you.
Yeah, thank you. Thanks for letting me talk, everybody. Appreciate it.
All right, Kristen, it was great to see you. Thanks so much. And thanks for going first. I know that's never a fun position to be in, is to go first. I hope you'll be able to stick around a little bit with us. We have a lot more speakers, but I appreciate your time. I know you're very busy and it was good to see you.
Nope, I scheduled all day today. I'm here.
Yay. Okay. Well, our next scheduled speaker is Ansley Ciego. I don't see her on. Ansley, are you on? Okay, I have not heard anything from her this morning, so I'm going to assume something came up. I know Pittsburgh got a lot of snow last night too. So if we can, I think I'll just go ahead and move on, and then maybe we can come back to Ansley if she's able to join us. Next on the agenda, I have Dr. Pamela, and can you please pronounce your last name for me?
Pupo. Hi.
Hello, thank you so much for joining us. She is at Marshall University, and today she is going to talk to us about plants that are foraged by 2 species of longhorned bees. Thank you so much for joining us, and the floor is yours.
Thank you. So let me see. Everybody can see my screen now.
Yes, I do.
Awesome. Great. Thank you. So thank you for having me. It is my pleasure to be here. I joined the society last year. I moved to West Virginia in 2020 in January, right before the pandemic. My main disclosure here is that I'm a botanist, but I started working with bees a couple of years ago when I was a postdoc at North Dakota State University. And so I'm mostly interested in the plants that these wild solitary bees use. And so before I start the talk, I want to acknowledge my team. So all of these people that are helping me with this project and making it possible. And so first is Jared Prasivka. He is a researcher with the USDA at the Agricultural Research Service in Fargo, North Dakota. and his postdoc Beth. They are the ones that maintain the sunflower plots and the ones that are collecting the bees. So I get my bees from them. Maddie Thompson, she was an undergrad working with me at North Dakota State about 3 years ago. And Rachel, she started working with me last semester on this project at Marshall. She's an undergrad at Marshall. And they have both helped me with some lab work, extracting DNA from the pollen of the bees or washing the bees and so on. So I just want to make sure I acknowledge them before I start. And so all of you know pollination is essential for producing fruits and seeds. So flowering plants, a lot of them are dependent on this moving the pollen from the male parts of the flower to the female part of the flower, the pistil, and they use different vectors for this. Many of them rely on insects for this transfer of pollen. In fact, 80% of wild plant species and about 75% of cultivated crops depend on insects for pollination. In the US, about 130 crops benefit from insect pollination, and some crops like almonds or palm fruits or melons completely depend on insects for this service. And so growers for centuries have relied on managed honeybees, which are generalists, right, as you well know. But honeybees have been rapidly declining. So they have been managed for centuries, and because of that, they have very little genetic diversity. And so that makes them very susceptible to things like changes in the environment, like changes in temperature and humidity, pests and pathogens. Because they have this little genetic diversity, they are more susceptible to diseases and pathogens and other things. Of course, they are very useful and easy to manage. They can just be transported to different locations to do the pollination services, but they are generalists, and sometimes they're not the best for the job. And so because they have been rapidly declining, our attention has been switched a little bit to wild bees and solitary bees that provide a free pollination service. A lot of these wild bees are specialists, and so they have co-evolved with specific groups of plants, and they are very good at their jobs. So they are more effective pollinators to many groups of plants. And so for this project, this is some preliminary data I'm presenting today. I've been working with 2 species of Melissodes bees, which are the longhorn bees. They are longhorn because the males have these very long antennae. They're very, very cute and very small. And they are sunflower specialists. And so you can see here a bunch of males just hanging out on a wild sunflower species. And so they are specialists of this group, the Helianthus species in the Asteraceae. And I have, as I said, I have been working on 2 species of these genus, Melissodes trinotis and Melissodes agilis. And so talking with entomologists, one of the first things I realized is that there is this big taxonomic dispute about whether or not these 2 species are the same thing or not. And so apparently they have these hairs in the posterior end of the animal that are different. And that's how they differentiate among these 2, or so I've been told. And so one of the first questions was whether or not these 2 are the same thing or not, right? And I'm a plant taxonomist, I'm not a bee taxonomist. But I said, well, you know, if we can study their foraging habits and maybe we can find some differences between these 2 species, that might help us with this problem. And so one of the questions, or another question I wanted to answer is, if these 2 species are specializing in different Helianthus species. So that's the first thing. Are they collecting the same thing or not? And then the other question is if they are in fact Helianthus specialists, or if they also forage from other different species, perhaps or species in different genera in the Asteraceae or another plant family. And so for this, as I said, Jared and Beth collected a bunch of these bees in sunflower fields. So the data I'm presenting today is coming from 1 sunflower field, part of the USDA fields in North Dakota. I have 85 bees for this data set. And so what we did is we start by washing the bees in the lab, and you can see these little tubes in here with all of the pollen at the bottom. So we wash them to make sure all of the pollen is out of the bee, and then we work with that pollen load. And so we have 85 bees or 85 pollen loads. So the pollen from each one of the bees is removed and put in a single tube. And then we amplified 2 genes, the ITS2, which is the internal transcriber spacer. It's section of the ribosomal nuclear ribosomal DNA. And then the tRNL, which is a chloroplast gene. Both of them are proposed as the kind of barcodes for plants. Plants are very tricky. It's not as straightforward as with animals. At CO1, the cytochrome oxidase 1 barcode works very well for most groups. Plants are not that straightforward, so there are a couple of markers that can be used as barcodes, and these are 2 of them. ITS2 has been used for differentiating within the Asteraceae genera and species quite successfully, so that's why I wanted to include that one as well. And so the process is very straightforward. I'm not going into too much detail about this. If you have any questions, you can ask me later. But basically, we remove the pollen from the bees. We put each one of the pollen coming from one single bee in a tube. We do the DNA extraction, which will extract DNA from all of those pollen, which are presumably different species and groups. And then we amplify just by a PCR. We cut these 2 fragments from all of that pollen load. And then we send that to next generation sequencing, which allows us to have a lot of data. And then we use these tags that you can see here. So each one of the primers we use has a unique tag that will allow us then to separate our samples so that I can know that this bunch of sequences come from that bee or that pollen. And so then we kind of reconstruct what plants were collected by each bee. And so these are some of the results we have. We see some surprising diversity of plants visited by a specialist bee. So this is, I'm showing you first the ITS2, the results from the ITS2 gene. And we see here the 2 bee species, Melissodes agilis and Trinodes. And so they, in fact, collect mostly Asteraceae. So here in blue, you see M. agilis collected about 80% of Asteraceae. M. Trinodes about 70%. They also collect quite a bit of Fabaceae, so legumes. We found some Brassicaceae, so mustard species collected by Agilis, and here a little bit of Linaceae by Trinodes. And when we zoom in a little bit into the Asteraceae genera for each one of the bees, we see that in fact, most of what they collect is Helianthus. So they are Helianthus species, sunflower species here. So about 2/3 of what Agilis collected was Helianthus. But they also collected other things within Asteraceae. So these Ratibi, the genus, it's the coneflowers. And these Solidago, it's the goldenrods. So we see Agilis collected a bunch of those as well. Trinodes collected mostly Helianthus. As you see in here, most of this is blue, but they also collected a little bit of these coneflowers, some of these goldenrods, but they also visited some other things like gumweed and ragweed and others. And these differences between the pollen collected by both species is statistically significant. And then when we looked at the other gene, the tRNL, we see basically a same pattern. So most of it was Asteraceae. They also collected, both species also collected a bunch of Fabaceae. But the interesting thing about this gene is that it showed us at least 3 groups of bryophyte spores, so mosses, moss spores. And I was just scratching my head and thinking, how is that a bee got spores, moss spores? So I was just, wondering about it. So we have a horn moss here and a couple of what you normally would identify as mosses as part of the pollen load in these species. And these differences at the genus level are also significant between the species, which means the pollen collected by both bees is different. So the pollen collected by Agilis and Trinodes is statistically different among them. And then I was talking with my entomology colleagues, and they reminded me that these are ground burrowers. And so they told me, well, maybe they are collecting some of these mosses to cushion their nests or something like that. And that's why you're picking up these spores in the nests, which for me, it's fascinating, right? And so our preliminary data supports that there are in fact differences in foraging habits between these two species. Of course, this is not enough to say if these are in fact two different species or not, but at least it's an indication they do have differences in foraging habitats, especially because they were both collected in the same habitat, right? They were both collected on sunflower fields, and there's still a difference in foraging habits. The other very interesting thing is that these data shows a much wider diversity of plants visited, even outside of the sunflower family. So yes, they are mostly picking up Asteraceae. Yes, they are mostly picking Helianthus pollen, but they're also visiting other things, even outside of Asteraceae. And for me, that was very, very interesting. And with this, I want to acknowledge some of the institutions that are making this research possible. The Insect Cryobiology and Ecophysiology Network at North Dakota State University when I was a postdoc 3 years ago, and the USDA in Fargo, North Dakota, and the University of Minnesota, the Genomics Center, where the sequencing was done. And thank you all for having me.
Thank you so much. That's really great stuff. It looks like you have several questions in the chat. If you want, we've got plenty of time if you want to go ahead and answer some of those.
Okay, let me see. Are the number of honeybee colonies really declining? Well, I think that's a better question for this group, since I am the botanist here and you are the entomologist. So all of the reviews I've read so far point towards a decline in these honeybee colonies, right? But I'm sure you are the ones that have the data more up to date. So I'm sorry, I'm kind of returning the question
Can
to
you
you.
stop sharing your screen so we can all see each other, please?
Sorry, yes.
Pamela, I am a beekeeper also. To the best of my knowledge, the bee decline is primarily due to mites. Because of the economic value of having bees for pollination, the huge amount of money invested in pollination, the beekeepers are producing more and more colonies every year. The annual mortality is skyrocketing, but the number of new colonies is also skyrocketing. And the number of colonies has been staying level for years, despite the massive mortality. The more important question, I work with viruses and mosquitoes. It's critical to know which animal a mosquito feeds on. So if you go into a chicken coop and you find the freshly fed mosquitoes and you test what they fed on, the vast majority fed on a chicken. Even if this species in the real world rarely if ever feeds on a bird. It's a mammal feeder. But because you collected them in a chicken coop, they fed on chickens. Because you collected these bees on a sunflower, the vast majority of pollen on that bee is going to be sunflower. It would be much more if you could have found some of the nests and collected bees returning to a nest and found out what they fed on at random, would have been so much more important. So you want to, when you collect bees for blood meal analysis, you want to collect them away from any particular blood source to find out what the mosquito normally feeds on. It's an interesting talk, but I would have expected lots and lots of sunflowers because the bees were collected on sunflower.
Yes. Well, that's a great point. Thank you. So I will definitely pass this along to the persons that are collecting the bees for me. So what we were thinking about doing next is serving larger sunflower plots. These are relatively small plots, and so they will naturally have access to other plants. So we want to see if this pattern holds if we collect them in the middle of very large sunflower plots. Maybe we will exclusively find sunflower pollen in them, maybe.
Can I ask a question based off of Michael? So you showed the ground that they make their nests in. Does anyone know, are they like leaf cutter bees that they like make the little partitions with?
Not to my knowledge.
So they don't put like pollen down there for their babies to eat?
I'm guessing they would. If they're ground nesters, they still have to find provisions. Now, I don't think they cut leaves for nest construction like leaf cutters do. So what they told me is that they
pollen in there.
Yeah, so what they told me is that they might pick up some mosses, little pieces of mosses to kind of cushion the nest, but I haven't opened a nest. So I really haven't looked at how it looks like on the inside.
It would be sad because you'd be like killing them, but I mean, that's science, sometimes it sucks. But like, if you dug up a nest and found like their little pollen ball that they mash in there for their babies as a larva to eat, that would probably be, but I don't know. It might be that they might do that only half of the time, right? Like, because they might forage and then they might collect something else for their young, but it might be, I don't know, there's so many questions, right?
Yeah, so sorry, another of the projects I have is with alfalfa leaf cutter bees. So I'm working with Megachile rotundata, and I am looking at the diversity, so the plant diversity in their brood provisions and in the nest. So I'm looking at both what they are feeding their larvae and what they are using to construct their
Okay.
nests.
Oh, cool.
So stay
I'm so
tuned.
excited. Yes, I hope you come back. I would love to know all about your stuff.
Yeah, I'm just, with the pandemic and the new position and everything, things have gone slower than I anticipated. So
Of course.
I'm starting to get these data out, which I'm very excited to be able to present preliminary data on this. I have recently started another project with Craig Larsener, I'm sure he's around, on wild ramp pollinators. So we'll see what we find in there too. So yeah, stay tuned. There are a lot of things coming up. Okay, sorry, I'm catching up with the questions on the chat. I have another one from Michael. Oh, I think this is what you already said, Michael. Yes, about collecting them in the sunflower fields. Okay, I have another one from Eric here. Do we find these bees far from sunflower fields? I really don't know. Again, I've mostly doing the lab part. I know these ones, these bees are traditionally seen as sunflower specialists. So they are mostly seen visiting sunflowers. And that's where these ones have been collected on. These have been collected on sunflower fields. I do have, so what I presented today is the data from one locality. I have bees from other localities near Fargo, including the landfill, for instance, so away from sunflower fields. I don't have the data processed yet, but again, stay tuned and we might hear about it later in another meeting. And I think I'm all caught up with the chat, I think, but if you have more questions, feel
Oh,
free.
just a comment more than anything else. This is Eric. Yeah, I always worry when I hear about specialists. It just makes me nervous when I hear that term. I used to work with some parasitic wasps, and we always try to figure out host range, and it's like, well, we never know, really. There's so many variables involved dealing with this aspect of things. It's a tough one, so I'm sympathetic on feeding ranges and everything. So,
thank you.
What was the time frame of your sampling? Because the information of the plant families, sometimes it kind of alludes to like different seasonalities. Was it the entire flower? Like, was it March to September, or was it just July to September?
Yeah, it was like 2 weeks in August.
Okay.
So there's really not much time for the floral composition to change.
Yeah, I have seen these longhorn bees a lot. I don't know if it's the right species, but I have seen them in the spring, like, because I go crazy for the spring ephemerals here, which I really hope you get to experience if you just moved. They're amazing, but I do see them flying around when I get really close to those flowers in the springtime.
Hey, Pamela, I have a question. So, those the bees in the family Apidae and Megachilidae, they have a kind of tendency of it's called the flower constancy. Once they visit one flower, they keep visiting the same flower over and over again. So in your data, I couldn't see the variation because everything is kind of in the bar. So I wonder, in the certain sample, you see consistently other pollens rather than sunflower, or?
So the other, again, not as abundant as sunflower, but the other, like the second family that we find in there are the legumes, the Fabaceae. So that's the second one that always shows up for these two.
Always shows up. Oh, okay. So is there any like samples like skewed toward something a lot or?
Within the Fabaceae, you mean?
Okay.
Is that what you're asking? If within the Fabaceae, so within the Fabaceae, we see trefoil, we see clover, we see a little bit of this.
Okay.
Yeah, but it's very interesting. So again, with this other data set I have on the leaf cutters, the leaf cutter bees, they do construct the nests. They're like a straw, right? So they construct one brood cell and then the other and then the other and then the other. But then we do see differences between the oldest cell and the youngest cell. And even though it's a span of a week, so the floral composition hasn't really changed, but their choice apparently is different because the composition of the brood provision of the oldest cell and the youngest cell are different.
Really?
So,
really?
Yeah, it's actually very interesting. Sorry, I'm just getting this data in, so I'm not able to present everything today.
Oh wow.
But it is. I'm planning on giving a seminar at the Department of Biology in Marshall in February, where I'm planning to present all of these bee data. So I will be sure to forward that information to all of you. So if anybody is interested in attending, it will be hybrid, so in person and virtual.
Okay. Yeah, because we have a research with the Osmia conifrons, which is like a leaf cutter bee, not leaf cutter bee, like mason bee.
Yep.
We never seen one row of, we always see one row of those like, or one tube of nest has always same pollen, always.
Interesting. Not in this case, and we are not sure, right? But in alfalfa leaf cutters, usually the first cell and the last cell, or the youngest and the oldest, are different sexes, right? So one will be a male, the other one will be a female. So maybe there is a relation with that. I don't know.
Right, but that's the sex of bees, but the kind of pollen is always same, in one tube.
But not for alfalfa, not for alfalfa leaf cutter bees. Yeah.
Okay.
Sorry, getting away from the long forms.
Okay, great.
Thank you.
We hear you.
Great questions. Thanks, everyone. And thank you so much for presenting.
Sure. Thank you for having me.
So we're going to circle back on our agenda. We had a little bit of a delay, but Ansley was able to join us. So I think we'll go back and have her present next. I'm really excited that she could join us today. She is the new curator of invertebrate zoology at Carnegie Museum of Natural History, which is right up the road in Pittsburgh. And she is going to talk about the evolution of beetle iridescence.
All right. Can people hear me okay?
Yeah, you sound good.
Yes.
Awesome, cool. Thanks so much. I apologize. Our schools were not canceled, just surprise remote this morning. So things have been a little hairy this morning over here. And Pamela, that was such a cool talk. I used to work in an alfalfa system in Australia, and it was a real trip down memory lane with the leaf cutters, which apparently are the absolute best at pollinating alfalfa. I will go ahead. Can I just go ahead and share my screen? Let's see if I can do this.
Green button on the bottom.
Okay, the button on the bottom. There we go. Share screen. All right, I am sharing this screen. All righty, cool. We've got beetles. Let's play the beetles. All right, great. So this is a talk about some of my research that I do. I'm actually a beetle taxonomist and a diagnostician for general insect things. So if you need help identifying a weird thing, let us know. That's a service that we provide for money as part of our support of forestry at the museum, but it's also something that we do for fun. So what I'm going to be talking about today is a little bit more flashy and exciting than taxonomy, and that is how iridescence has evolved in beetles. So Iridescence is what we call a structural color. It's a type of structural color. So the colors in our skin or in our clothes or paint, or what we call pigment colors, that's colors where you have selective absorption of photons by a particular pigment or a chemical. Like in our case, that would be melanin or in our skin. Structural colors arise from photonic interactions with the actual structure of the cuticle. So the sort of the nanoscale structure of the actual exoskeleton. And so when you see a color that lasts in an insect specimen way longer than it should, like if you have something like a ladybug or even some monarchs, after you pin them, the colors will tend to fade over time frequently. With structural colors, those colors will persist indefinitely. And of iridescence, we define that as a subclass of structural colors where the color we perceive tends to change as we change the angle that we view it at. That's just the working definition of iridescence for people who work on this kind of photonic biology. And there are a couple of fairly common ways that beetles can make iridescence. This little diagram down at the bottom of the screen here shows you sort of an example of pigment colors contrasted with some of the structural colors and iridescence types we're going to talk about today. The most common is probably things like multi-layer reflectors, if you've seen a dogbane leaf beetle, or an agrylus, if you're working in forestry. This should be really familiar to you. These are the kind of metallic colors. And what's going on with these guys is you have a stack of layers of chitin that happen to be at the exact right thickness that's about 1/4 of a particular wavelength of light. And when they're that thickness, they're going to reflect that color, that wavelength of light, with what's called constructive interference. In other words, they reflect it super, super brightly and super strongly. And so it gives us this impression of really, really vivid colors. And so it's the thickness of the chitin layers in the exoskeleton that's going to affect what color you perceive. And this is what it actually looks like when you slice it. It is a lot harder than you might think to take a nice, neat little slice of beetle cuticle. It's very, very tough. It tends to split. But here you can actually see this nice cross section of those layers in the chitin of this little tortoise beetle, Aspidomorpha. So this is probably your most common type of beetle iridescence that you see in nature. Another kind that I'm a big fan of personally is diffraction gratings. This is when incident white light hits a series of parallel slits or ridges and gets diffracted or scattered into its constituent wavelengths. And you'll get this kind of rainbowy sheen. On the right of this slide, if you guys can see that okay, is a little section of the reflectance from a diffraction grating on a ground beetle called Selenophorus. I call this spectral iridescence because it produces an entire reflected spectrum. And this does seem to have a little bit of an anti-friction function as well. You see it a lot in beetles that are crawling around in mud or decaying fungus or other kinds of gooey substrates where you might have a problem with friction with sticking to the substrate. And you also see it a lot in snakes and in small burrowing lizards, like little skinks. You'll see this as well. And let me see here. There we go. This is an SEM of some diffraction gratings from my favorite group of beetles, the round fungus beetles or truffle beetles, the family Leotidae. And if you look at this lovely little eglantine over here, you'll see that it has this beautiful, vivid green and blue spot on the elytra. And that is just a patch of spectral iridescence coming from a diffraction grating on the elytra. These are just little ridges. They're not hairs, they're not seedy. And it's much more likely that they have a function in shedding, in preventing friction or shedding water than they do as some kind of visual signal. The other kind of neat thing about these guys is that they're not really supposed to have colors at all whatsoever. The vast majority of them are little, round, and brown. And so it's extremely exciting when you find one that has these really interesting, vivid iridescent colors. Now, those are the two main ways that we see iridescence in beetles, that it seems to be common. However, there is a third beetle iridescence mechanism that I'm going to focus on today, which I am super excited about. This is what we call three-dimensional photonic crystals, and it's a real mouthful, but I think it is pretty cool. When I say three-dimensional photonic crystal, here's what I mean. A photonic crystal is just any structure that interferes with photons that either reflects them or transmits them or blocks them or what have you. So a one-dimensional photonic crystal would essentially just be that multi-layer reflector that gives us those metallic colors. Two-dimensional photonic crystal, these are rare, but they do exist. We see them in, if you've ever seen a picture of the bright blue melalonthine scarab from Hoplia cerulean, I think it's called, from Europe. Those guys have a two-dimensional photonic crystal. A three-dimensional photonic crystal is a structure that has 3 dimensions of periodicity, or 3 axes along which it is periodic. That periodic structure is what enables you to get constructive interference when you're reflecting a particular color. So this was first described in Pachyrincine weevils. These are a lovely little broad-nosed weevil from Indonesia and Papua New Guinea and the Philippines in 2003. And it was described as an opal weevil because, and this author, Andrew Parker, made a little imprint of the structure inside the individual hairs on the weevil that was giving it these colors and said, oh, this looks a lot like the microspheres of silica that we see inside the mineral opal. So if you've seen a gemstone, the gemstone opal, he said, oh, this looks just like that. I'm calling it an opal weevil. So what that actually looks like, if you zoom in, these little kind of, what would you call these, forbidden mentos, these are the scales of one of those opal weevils. These are the scales of a Pachyrincus weevil. And if you take one of those little colorful discs and chop it in half, what you will see is this extremely complicated little 3-dimensional photonic crystal. And as you can see from the image of Lamprocyphus there, the crystal domains are oriented in different directions. So there'll be a chunk of a crystal pointing this way, another chunk of crystal next to it pointing another way. And because they're variably oriented, you get direction independent structural colors. So with most iridescence, think of a morpho butterfly. As you change your angle of observation, it's going to change the color that you perceive. But with these guys, you get angle independent, really bright saturated colors because you have multiply oriented little crystal domains. And it turns out that there's some actually really sophisticated photonics and physics going on inside these tiny little weevils. There's a recent paper that just came out that I'm a big fan of by one of my earlier co-authors, who found that if you compare the structures of the photonic crystals inside the scales of, for example, this Pachyrincus here, you'll notice in the macro photo that you've got it's almost a whole spectrum, right, of red, orange, yellow, green, blue. They referred to this as a tunable photonic crystal because all you have to do is slightly alter how big the air spaces are within that sort of spongy crystalline matrix, and that's going to have a huge effect on the color that's produced. And that's a lot easier to change your color. So if you're a bug and you want to change your clothes from an evolutionary standpoint, usually you would have to sequester pigments from a plant that you're eating, perhaps. A lot of hawk moth caterpillars will do that. Or you will need to synthesize the pigments de novo in your body. You'll have to metabolize them. That takes a lot of energy. But if you have structural colors like these photonic crystals, it's comparatively easy from an evolutionary perspective to change your color, to go from being, red to blue to green. You can also, this is also true of multilayer reflectors as well. But the photonic crystals are really interesting because it's such a sophisticated, complicated mechanism for manipulating light. They're also kind of neat from the perspective of engineering. There's a field of sort of a subdiscipline of engineering called biomimetic engineering, which just means stealing ideas from nature. And so there's a really strong potential to, if you can make these really bright colors using just chitin, the polysaccharide that bugs are made out of, and have these crystals that seem to assemble by themselves. They're lightweight, they're non-toxic, they're biodegradable, they're, flexible. Think of the things that we could make with that. Think of being able to make a, beetle-based visual display, a billboard, a cell phone screen. There's a lot of really neat potential in there. So the question that I had is, how do these crystals actually evolve, and how do they form inside the weevil itself? And I apologize, but I am actually going to continue this talk while I walk downstairs to plug in my computer, because apparently I forgot to bring my computer plug upstairs to my office. So this is the question that we sent out to ask: how did they evolve in weevils? and in beetles themselves. So the first thing we did, because we're taxonomists, is we sent out to survey the taxonomic distribution of these crystals. And we looked across all of the weevils. We even looked outside the weevils and the rest of the group Phytophaga. That's going to be the leaf beetles and the longhorn beetles. This research was performed with the Australian National Insect Collection, which has a really respectable assortment of weevils. And so that was a pretty good place to start. Once we had surveyed across the entire diversity of Phytophagan beetles for structural colors and then see what their mechanism was, we used both electron microscopy, so scanning electron microscope, and short angle X-ray scattering to characterize the internal photonic structures that are inside those scales. And this is just a little tiny diagram that I made of how short angle X-ray scattering works. Essentially, you take a very, very high powered X-ray beam, a beam of accelerated particles, and you blast it through that little weevil scale, and the pattern that you get that the X-rays make when they get diffracted and scattered through there tells you exactly what kind of internal geometry there is in the photonic crystal. So this research was done down at the Australian synchrotron in Melbourne, Victoria. There's a picture of somewhat younger me being drunk on my own power in front of 8 computer monitors at once. And this is what the actual output data looks like. You see on the left the diffraction pattern of how the X-rays have been scattered as they pass through an individual iridescent weevil scale. And those little spots that you see in the symmetrical scattering pattern correspond to the symmetry peaks in the graph on the right. And then once we had that data to characterize the photonic crystals, we could map what we knew about those photonic crystals onto a phylogeny of the group, reconstruct the ancestral states, and figure out the exact history of how they evolved. So what we found out is that the ancestral condition for photonic crystals and beetles seems to be, at least in weevils, seems to be this sort of spongy matrix that scatters white light. This is really great if you are, like this beetle is, a little brown fungus feeding beetle, and you want to be cryptic on brown fungusy wood that has lots of white sort of mycelia or hyphae or fungal fruiting bodies on it. It turns out this is a great way to be cryptic. So we think maybe this is, not only is this the ancestral condition, but maybe this is a potential evolutionary pathway. The next type of photonic crystal that we found that's actually organized and produces an actual structural color instead of just sort of a white appearance is a simple sort of hexagonal network that you see here. Let's see, there we go. And we also saw diamond type geometry as well, another diamond type network. And then finally, the most sophisticated kind is a gyroid network. This is all geometry terms and you don't need to remember for anything, but there's a surprising geometrical diversity within these individual photonic crystals from different species. We also, somewhat to our surprise, found that there were 3-dimensional photonic crystals in longhorn beetles. They are in lamine cerambicids, and just like in the weevils, they are inside the individual scales. So those are just modified hollow CD. And these guys have a different kind of geometry. They have a cubic geometry instead of the type that we see in the weevils. And it also seems to be, instead of sort of the air spaces, you seem to have these sort of nanospheres. I love this particular lamine. I think that's a Prosophocerolactator, and it's got what we would call quasi-ordered nanospheres. This is exactly the same thing as when you put zinc sunscreen on your face. It's just a bunch of little nanospheres that are super good at reflecting white light. We also see what have been described as colloidal crystals. This is in a longhorn genus Anaplophora, and they don't even seem to be organized. They're simply this assortment of quasi-ordered nanospheres of chitin, and this is absolutely fascinating. And one of them, this is a paper that was published in 2009, is a cerambicid that seems to have secondarily rearranged its 3-dimensional photonic crystals into a multi-layer reflector, like the one that we see in your other sort of basic buprestids and other kind of metallic shiny beetles. Now, this structural diversity spans a whole lot of different types of geometry, right? So if you're a physics person, an engineer, a crystal person, you would look at that and say, oh, well, look at all these, cubic and gyroid and hexagonal crystals. But if you are a chemist person, like an organic chemist, you might look at that and say, hey, those actually all have something in common. Those are all geometries that you can make from lyotropic phases. Those are all lyotropic phases. And I know that phrase means nothing to probably everyone. A lyotropic phase is a word for a structure that you can create by taking two molecules, an amphiphilic molecule and I should say just taking amphiphilic molecules and a polar solvent and combining them together and letting them separate back out. So imagine you have salad dressing, you have some balsamic vinegar, you have olive oil, you shake them up together. And then if you set it down, that homogenized dressing is going to separate back out. And as it does, it will form structures like this on the way back to separating back out. That's all we mean by lyotropic. This means we have an idea of how these crystals could actually be forming inside the Weevil scales during pupation. We know they're formed during pupation. We know that they are in place by late stage pupae because we have cut open lots of Weevil pupae trying to catch them in the act of forming, and they are almost always done by the time you will have a pupa that's ready to hatch. Which is really exciting. So it really seems like this has to happen in an aqueous environment. And sure enough, we only see these crystals inside closed scales, inside a hollow scale. You don't see it on the surface of the elytra. You don't see it if you cut open the cuticle. You only see it on the inside of a hollow watertight structure. So it seems like there really is some kind of an aqueous environment that allows lyotropic phase separation to take place. Finally, where do we see these structures across beetles? We don't see them at all in the chrysomelids, the leaf beetles. They appear in two different tribes of cerambicids. And then in weevils, they are all over the place. And so you might look at that and say, wow, it must have evolved so many times in weevils. Look at all of these different weevils that have three-dimensional photonic crystals. Incredible. Well, we know it evolved at least twice because the cerambicids and the weevils are widely separated enough, and we don't see it in the chrysomelids, that it seems like that's probably at least two different origins. However, when you put the three-dimensional crystals that we've just described, when you map that onto a phylogeny of weevils, it looks like they all belong to a single group, the entomine weevils or the broad-nosed weevils. Every single weevil that we found three-dimensional photonic crystals in all belongs to this clade. So it looks like there is actually just one origin within the weevils. And that's why it's so important when you're asking big evolutionary questions to take phylogeny into account. You might look at this data and say, oh, wow, this is so common and widespread within this family, but actually it's all within a single clade, which means it probably evolved only once within this family. So putting all of that information together, that lets us come up with a hypothesis about how these ridiculously beautiful iridescence mechanisms evolved in weevils. It looks like we have an ancestral condition where the hollow lumen of the scale is filled with this spongy reticulate network of chitin that scatters white light and gives you a little bit of an advantage with regard to crypsis when you are a little fungus feeding beetle. If you modify that, if you tighten that up a little and get a slightly more regular hexagonal structure inside your scales that coincidentally happens to have evolved right at the same time in the same clade that became specialist feeders on angiosperm plants, on leafy green plants, you have this bright green that's a really hard color to synthesize if you're a bug, especially a beetle, and that happens to give you this crypsis that helps you blend into your substrate. And then with a few additional geometrical tweaks of those photonic crystals, simply by altering the amount of solvent and the amount of your amphiphilic chitin molecules inside the lumen of that scale, you can get these more sophisticated, incredibly brightly colored gyroid and diamond-like crystals. We have no idea what they're for. Some people think that it's a way to be aposematic. You can use them to mimic other species. There are certainly some weevils with three-dimensional photonic crystals that use them to mimic ladybirds and other chemically defended beetles. But we think some of them, the pachyrinkines, for example, are one of the notoriously hard to pin types of beetles. It could be a way to say, don't even bother trying to eat me. I'm way too hard and crunchy. We don't know. That's a mystery still. But that is our hypothesized evolutionary pathway for how they've evolved and how they form within the scales themselves and what the necessary prerequisites are to evolving these photonic crystals in the first place. So that is my little side project research that I'm really excited about, but I would also love to talk to anyone at any time about beetles if you have a beetle question or beetle facts or any other stuff like that. So thank you so much for letting me slide in late and blast beetle information at all of you guys. A couple of little fun, I'm just going to throw in a couple little fun things that you can do. People have found ways to synthesize these. We are making synthetic photonic crystals out of chitin using the same formation principles as the beetles. And these are just some photos of what we've got there. So there you have it. And I am going to stop sharing my screen.
Thank you so much for that presentation. I'm so glad you were able to join us. I know today was really chaotic for everybody, like all the snow and they canceled school and it gets a little nuts. So I really appreciate you making time for us. Really fascinating. Do we have follow-up questions from anyone?
Yes, I have a question.
Oh.
Hey, thanks for the fantastic talk. So so based on your presentation, so you are suggesting that outside Coleoptera, all of those metallic bodies like hemipterans, dipterans, hymenopterans, they are just a normal structural color.
They are probably, they don't seem to be photonic crystals. If the structural color that you see, the kind of iridescent effects, like you would see in like a scutalarid or a blowfly or one of those other, iridescent insects, those all seem to be coming from the multi-layer reflector in the cuticle. So it would be rude of me to suggest that that's a boring type of structural color. I just happen to have been studying the photonic crystals when I'm not doing taxonomy and phylogenetic stuff, but there are a few exceptions. We do see three-dimensional photonic crystals like that in some groups of butterflies as well.
Okay.
So and then there are some bees, some of the blue-banded bees in the genus Amygilla. Those seem to have a two-dimensional photonic crystal, but not a three-dimensional one.
Oh, okay, great.
So,
So
Hey,
how many people are working on these things? I just wonder in the world.
lots, tons of people are working on it. There's a whole group of people who work on structural colors and photonics and what we call visual ecology, which is why does animal make color? What is it for?
Okay.
There's a beautiful paper that just came out looking at convergent evolution of transparent wings in Lepidoptera. So if you've seen a clear winged sphinx moth, that's evolved a whole bunch of times, and they looked at it mainly in butterflies, and that's just a wonderful, wonderful paper. The problem is all of it is multidisciplinary. There's the engineers who are trying to copy the photonic mechanisms that the bugs have invented. There's the bug people who just want to know whether or not they can use these characteristics to identify species. There's the ecologists who want to know what advantage that gives you in different kinds of environments, or whether you can, make soup. Like, for example, there's a great paper that came out 2 years ago, I think. If you've ever seen an eyed click beetle, Aloes oculatus, big gray click beetle with two big black eye spots on the back of the pronotum, it turns out that is a form of structural color. Those are little kind of, the technical term is a nipple array, but it's almost like a shag carpet or like velvet. It's all of these tiny little spikes packed in closely together. And it's so good at absorbing light that when you sputter coat those eye spots with gold to take an SEM, they still look black.
Oh.
So you cover the surface with gold and they still look black because the structure is what absorbs photons and doesn't let any escape. So there's just, if you find a bug with a weird, color or eye spots or signal or mimicry going on, there's almost certainly some really cool physics explanation behind it.
Great. Good to know. Thanks.
This is Eric from DHHR. Have any of the descendants of any of these 3D photonic crystal weevils ever lost it? Have we seen a shift where they actually lose that bright color over time as a species, as a genus, I guess? Any of the descendants?
That's a great question. There are some where they've lost kind of the iridescence. So if you look at some of these species, especially with the photonic crystals, they'll have this kind of sparkly, glittery look, almost like they're wearing glitter And there are weevils where the surface of the scales has been modified so that the colors are matte instead of kind of glittery and sparkly. And we see this only in weevil species where they're trying to mimic something that is toxic. So trying to mimic a chemically defended beetle like a lysid or a coccinellid. So they've certainly lost the sparkle. There are a few species within things like Pachyrhynchus where they have sort of lost the scales. They've just decided not to have those bright colors. Why? I don't know, but yeah, it has been lost.
Well, the
sorry,
first one sounded like they were shifting their behaviors, or somehow they modified their behavior. Somehow their behaviors were modified according to the shift in their physical structure as well. It sounded like in the first one. In the second one, it's unknown. So
yeah,
that's kind of neat. Thank you.
it's really neat. I see I have a question in the DMs that I will answer right now. Some small spiders in the family Theridiidae are highly iridescent. Do you know the mechanism there? And this is a whole other paper, but Theridiids and some Tomicid spiders as well have lovely structural colors that they make it's a multi-layer reflector, but it's made of guanine crystals. So the layers in those cases are made of guanine crystals, and they often have these lovely sort of silvery or iridescent colors. Oh, I see a kitty cat. My cat radar is highly attuned. So spiders actually are one of the few arthropods that can do reversible color change. So it's, we think of chameleons, right, as the peak example of things that can change color back and forth repeatedly. But there's actually a number of arthropods that have been able to do that, and spiders are one of the most notable ones. That's how things like crab spiders can match their substrate. So I think that's it for questions for me. So thank you so much for letting me yell about beetle iridescence.
Thank you so much. And again, I really appreciate your time. Those of us in Morgantown are fortunate that Pittsburgh's only about an hour and 15 minutes. So I know I spend a lot of time up there and a lot of time in the museum. So it's really great to have you be a member of the group. And several of you maybe got to meet Ansley when she came down to Watauga for the summer meeting. So one of the things we'll decide in the business meeting today is where we're going to have that this coming summer. So.
And I just want to jump in really quick. I apologize, Rachel, but I want to say to everybody who's in this meeting, you are welcome to come up to the Carnegie Museum anytime. Unlike other museums, Smithsonian, we are shut down for the pandemic. You can come look at specimens. You can come bring us stuff to identify or deposit. If you do accession vouchers, we're always happy to take those too. I know that you guys have your own insect collections in West Virginia as well, but the museum is open for business, and I'm relatively new here as well. So I'm happy to meet new people and welcome you into our collections. If I can also say one other thing, I apologize if this was brought up in the beginning of this meeting when I was not here. If anyone knows, John Rollins was my predecessor at the museum, and he died the day after Christmas this last year.
No.
And we are in the process of wrapping up his assorted loose ends, and we are also collecting, if anyone was friends or collaborators with John, if you have a story or some fieldwork photos or anything like that, I would be really, really happy to get those by email. We're in the process of arranging a memorial for him. So. That's all.
Thank you so much. That's sad news. John was probably my first contact up at the museum, and he was such an open guy, and he was quick to, come show you the collection that he was so proud of, and such a fantastic man. So we're sorry to hear that. I'm sure, I know I have a few stories I'll email you, and maybe others as well. So that's a loss to the entomological community as a whole, really. So we're really sorry to hear about that. Well, again, thank you so much for joining us. I'm gonna just keep going with presentations if we're okay with that plan. We've been at it for about an hour and a half. The joy of virtual is if you could, just keep yourself muted, and if you need to run and go get coffee or do whatever you need to do, I think we'll just keep on moving on with our presentations. So next up we have Dr. Park, who, for those of you who don't know, is at West Virginia University. and he is going to talk to us today about spotting insects from the sky. Dr. Park.
Okay, thank you. Can you hear me and can you see the screen?
Yes,
Yes,
we
it
can.
looks good.
Okay, thanks. So I'm gonna detect some insects with the 3D platonic crystals using satellites and drones. I think that's what I should do, right? So, my project has been focusing on like how to detect insects or insect-associated damage on the crop or forest using aerospace technologies such as satellites and drones and airplanes. So the thing is like this: it's just like looking for a needle in a haystack. You know, the challenge is there are lots of haze. It's so time consuming, and that's why we need a lot of resources. So that's why it's challenging and it's very hard to do. It is the same as actually the invasive species came here. So basically, so this is one of the invasive species like emerald ash borer. I think it has like a two-dimensional structure, right? Color. So we have a lot of ash trees to check. And then it's so time consuming and sometimes dangerous. And that's why we need a lot of resources, and then we don't have a lot of resources. That's why it's very challenging. So if you think of, oh, where are the invasive species? And somebody has to go out and look for it, right? In the United States. And even we don't know what kind of invasive species are here or will be, right? So it is very challenging. So by doing ground survey, it is very limited, or sometimes it is almost impossible. So can we use some technologies to overcome or help the shortage of resources to detect invasive species or other insects? So whenever we talk about technologies, aerospace technologies, these are the technologies we are talking about. We have the technologies in the space or in the air or close to the ground. So today what I'm going to talk about is more likely satellites and some aerial photography and actually drones. You know, each method has a pros and cons. So satellite, we can cover large area in a very short period of time, but the problem is the low resolution of images. And the other way, drone is very close to the ground, so it can provide high resolution image, but it is impossible to cover entire United States in a day, right? So there's a pros and cons. So that's why we use multiple technologies together to find whatever we want to find on the ground. So in my lab, actually, we started drone project about 2007. And you know, we started with the flower detection because flower is not moving, it has a color. And now actually we are using artificial intelligence and machine learning to automatically detect the flowers. This is related to our the pollination project. And then later, you know, we started detecting insects on the ground or on the plant using drones. And now actually we are using multiple sensors to do that. And then we started a satellite image analysis to detect insect damages. And again, you know, now we are using sensors. And the bottom line is now we are using artificial intelligence and machine learning for pest detection, including insects. So this is what my lab has been doing. So today, actually, I'm going to talk about only kind of insect related, but start with flowers so that I can show you the kind of the procedures. So on the left, this is actually the California vineyard. It's, you know, like a lot of grapevines in there. And in early 2010, there was a outbreak of morning glory in the vineyard. So morning glory actually to prevent harvesting and, you know, other management issues. So growers want to detect these morning glories and then cut it off. So what we did was, okay, these are the morning glory flowers. And then we used actually, we call the fixed wing airplane drone. So it's just like an airplane. And this is about 2008, 2009. So we didn't have a good drone at the time. So this drone has a camera on the bottom, and this is a styrofoam-based drone, so it's very light. And then this was the kind of general scheme. So the drone takeoff, fly serpentine pattern, and return back to land. And then during the flight, it takes like a series of aerial photographs, and then later we stitch them together. So that's the kind of drone operation plan. So you can see camera, 10 megapixel. It was the highest at the time, 2008. 10 megapixel was kind of very high resolution. And it's a GPS. So whenever drone takes a picture, we know exactly where the photo is located on the ground. So this is kind of a very quick view of our image analysis. So the A is the original image from the drone, downloaded from the drone, and B is kind of enhanced. So now you see some of these purple colors, those are the flowers. And C is actually showing the putting filter on the image. So, purple, the opposite color of purple is kind of cyan. So if you put the cyan, so this purple color shows up, and then we put the command, okay, make purple white and everything else black. So in the D, you see white dots. Those are the morning glory flowers. So we can cover this one in a large area, again, short period of time. That's the idea. And also when we did that, there's a problem because there is other things in the vineyard. So, it is not morning glory flowers, but it showed as morning glory flowers because of those shade and other object in the field. So we did additional analysis. It's called the shadow remover technique. So basically we take out only where the vineyard or grapevines are, and then analyze it. And this is the final products. So having this, we can find all of the morning glory flowers in like a 5 mile by 5 mile vineyard. So, and then we helped growers that way. So this is kind of done about already 10 years ago. So actually after this, we were thinking that, can we detect the insects on the ground? And then if we can do it, it'll change the game in entomology, right? So we tested. So this is about 1 meter by 1 meter bed, plant bed, and then we put some plants, and then surely we put some insects on it, right? And then fly over and take a picture. So this is actually the original picture look like. This is about 45 meters, 50 meters above the ground. And this is a glove. And this is a 1 meter by 1 meter plant bed with insects embedded. And then, we enhance the image again. And then you see now actually 6 different butterflies. And if you are a butterfly expert, I think you can say roughly what the species are. But the problem is we had about 20 more insects in this 1 meter by 1 meter plant bed, which include flies, beetles, and we couldn't detect it. It's too small to do. Yeah. So it was about, again, 10 years ago. So now actually 10 years later, so we have a better cameras, better sensors, and better drones, and better actually analytic methods. So this is actually what I'm gonna present today. So can we detect actually moth pupa? We choose pupa because it doesn't move, right? So we have a drone here. So it has multiple cameras called the multiple sensors. And also we have, this is autopilot, which means we don't fly, we program it. And then the drone goes certain place, certain altitude, and then do certain things like take a picture, take a videos, and that kind of things. So this is my actually sabbatical work in Asian countries. So this is like a saddleback moth. So basically, saddleback moth is problem in the like ornamentals, and especially like a roadside trees. And this is the larva. And then when they become a pupa, it looks like this. It's about what, 3/4 inches and 1/2 inches. And this is the pupa. And this is the time the people go out and scout it and sample it so that they can make a recommendation for pesticide application in the following spring. So if they see a lot of these pupa, and then they actually they decide to apply pesticide in the following spring. So anyway, these are the pupa, and this is what we targeted to detect it. Again, one more time, this is about 1/2 inch, 3/4 inch. So this is a video clip, and I'm gonna show you. So drone is flying down, and so, again, you know, this saddleback moth is the pest of this, actually the ornamentals. I don't know you can see it or not, but so this is actually the ground view, okay? And then this is these are the pupa. And on the right, can you see that this one is kind of open, which means the adults moths already came out. So this is not this year's pupa, this is the last year's pupa, okay? So this is actually the drone shot. What you can see is these white things, those are the pupa. And if you zoom in, now you can see all of this. Can you see that? So these on the right top, white pupa with the black opening. So which means these are not this year's pupa. This was last year's pupa. So one of the problem they have is whenever they actually count the number of pupa from the ground, they cannot see this exit hole. So they always overestimate number of pupa because they couldn't distinguish this year's pupa or last year's pupa. But if you see from the sky, you see these actually open pupa, which is last year's pupa, and then unopened one may be more likely this year's pupa. So it actually estimates more precisely so that it can reduce actually unnecessary pesticide application following spring. So this is actually what we found. So x-axis is the actual number of pupa, actual number from the census. And the y-axis is number of closed pupa, okay, by visual ground survey. And this one, the solid line, and then drone survey, this is actually dotted line. So what it says is visual ground survey always overestimate number of pupa, and drone is underestimate because drone cannot find all the pupa, right? But it's very close to the actual number of pupa. So if this is possible, can we do flying insect? Can we detect flying insects? So this is actually the mason bees, the Japanese home-based bee. Actually, that's what we studied. And so the idea is when insects fly, they have to use a lot of flight muscle in the thorax. And then whenever they use muscle, the temperature goes up, right? And then actually the temperature can go up very high, up to like a human body, like 90 degrees Fahrenheit, okay? Or 30 degrees Celsius, 26 degrees Celsius, right? 36 degrees Celsius, okay? So anyway, so if we can detect that heat from the flight muscle on the thorax, we may be able to detect the flying insect. So this is actually our thermal camera, and so you're gonna see it. So bee is flying in. Okay, here we go. So can you see this is a fireball? That's about 31 degrees Celsius. And this is the bee, flying bee, right? So one more time. I don't know, I can do one more time. Oh, okay. So bee is on the outside, now actually going to the nest. And now you can see this fireball. Because flying insect has a high temperature on their thorax, that's why we could detect it. Okay, so this is kind of one of the shots I can share with you. So A, there's a bee on the flower. but we cannot detect it because this bee is not flying, right? And then C and D, so there's a bee here on the flower, but it shows like a very high temperature because it's flying. And then E, can you see bumblebee? So in F, this is what flying bumblebee look like in a thermal camera. So it looks like there's a trace on the B, D, and F. This is called MXD technology. So basically, if you use 2 cameras, and then one is normal camera and the other is a thermal camera, and you can overlay. And from the normal camera, they trace out the object and then put onto the thermal camera. So that's why you can see this kind of trace. Yeah, so anyway, my point here is if insects are not flying, we cannot detect it with thermal camera, but if they're flying, we may be able to detect it. So that's the kind of big thing. So this is actually another shot. So these are actually the nest of Japanese hornface bee, the mason bee. And this is actually right, is a thermal camera, thermal image. So what you see here is you see 6 bees are in the nest. So these are 6 bees, 1, 2, 3, 4, 5, 6. But if you look at the left image, you may not be able to see the bees because the bees are inside the nest. So like this one, like a bigger dot, which means bees are closer to the entrance. So that's why we can detect more hits. So, okay. So if we can detect the bees, flying insects and, flowers, so can we detect all other like insect associated plant or tree damages? It should be very easy, right? So this is actually the image of 4 webworm, and this is the thermal image it looked like. So because this 4 webworm kills the like a plant tissue, like leaves, and when you take this thermal picture during like noon to 2 p.m. when the sun is high and the temperature is high, these actually the 4 webworm nest observe the heats. So if you look at on the right side, this is about 6 degrees Celsius higher than all other background. If this is possible, we can put this sensor, right? The thermal camera and the drone, and we fly over and detect all of these 4 webworm nest. So that's the kind of idea we are going for in terms of pest detection. So even actually, if we download the image and then we can go very detailed about each branches or leaves, right? So if you look at this branch observe more hits, which means this branch may be dying compared to other healthy branch. Okay, so that's the kind of idea. So if this is possible, and then we have been using like a thermal sensors and normal camera, but can we put other sensors? So, or can we do very like a large scale, like a damage assessment of insect outbreak? So again, this is from my sabbatical work from Asia. So in South Korea, there was a problem with the beet armyworm in the cornfield when I was in there. So this is a beet armyworm, and they've defoliate the corn. And on the right, so these are 3 people, and it looks like it's not planted, right? But it is not. It is planted. But this beet armyworm came and then completely defoliated this area. So these are the more area forests. So basically, A and D is like early and late in the season, right? So it's no damage. B and E, you can see like a damage already. And C, it looks like they haven't planted yet, but no, this beet armyworm ate all the stems as well. So it looks like empty. So that was the problem. But the problem, one of the biggest problem they had was the area is very like large, like more like a Monongalia County, Mon County size. So they couldn't do quick assessment of damages. So that's why drones kicked in for this project. So the area survey plan was, drone flies based on autopilot path, right? And then download the image, align and stitch the image, and georeference the image, right? And then do the image analysis, and then estimate defoliation. So that's kind of the survey plan we had at the time. So this is the area mapping. So this is, again, we are not flying, we program it and then command the drone to go and do the survey and basically take a picture, right? And also we did the low altitude validation mapping, which means they fly very low and we want to make sure the damage is done by defoliation, not others, right? So it flew very low. Again, this is also autopilot, the program. So this is the area mapping. This is one of the shot on the A on the left. This is one of the image and then showing. And then on the right is one of the image for validation mapping. So what you see here is, again, this drone fly very low and you can see defoliated the leaves and then, the feeding signs. Okay. So this is actually the what we end up with. So the red means this is the undamaged the plants, okay, with the image analysis. And then it turned out that in this area there were rice field, paddy rice field. So this big army one don't eat rice. So that's why all of these red, dark red area, this was the paddy rice field. Makes sense. And then we command again, then you automatically calculate the area of each block and then damaged area and percent damage. So this was done in 40-minute flight and 1 hour image analysis. So this is actually the great way to do the large scale area survey, right? For insect damage. So this is using just normal image, right? But what about, there are some green structures. It looks like, the field in terms of color, crop field in terms of color. So we couldn't distinguish it, right? So we used the different sensors. In this case, we put the near infrared sensor. So basically having near infrared sensor, we can calculate, it's called the NDVI, normalized difference vegetation index. So the idea is if the plant is healthy, they reflect 50% of near infrared color and 8% of red color. That's the kind of healthy plant. But it's damaged or it's senescing, and then they reflect less near infrared color and more red colors. So based on this NDVI index, and we can judge if this plants or crops or trees are healthy or damaged or senescing. So this is actually the drone we used. There is a normal camera here, but at the same time, we put another sensor here. This is called near infrared sensor. Drone is about what, $700, $800, but this near infrared sensor is about $3,000. So that's kind of where it goes. So this was done in Orchard in West Virginia University. We have a Cornisville Tree Fruit Research and Extension Center. So there is an apple orchard, and this is actually the flight path. Again, this is autopilot. We program it and then drone go and fly over. But in this case, they have two sensors, right? Normal camera and near infrared camera. And we download the image and we stitch it, right? And then, so this is actually the RGB, normal camera. So background is a GIS map, and in the middle, this part is actually RGB images from the taken from the drone. And this is a near infrared image from the sensor, and this is a red color sensors. Now, actually, if we have these near infrared and the red wavelengths, which means we can calculate NDVI. So this is an NDVI map. So now what you see here is green means healthy vegetation, and red means unhealthy vegetation or no vegetation. Okay, so if you zoom in here, it looks like this. So you can see individual trees, and this tree is very healthy, right? And this tree is a smaller or it has some problem, right? So now you can actually judge individual trees by doing this. Okay, so this is again using different sensors, and you can have a different information. So right now, actually, if this, so far so good, but if we have a different field, we have to do different analysis every time, or different sets of data we have. So what we try to do is, can we train this data with machine learning so that next time when we fly over, we don't have to go through all of these image analysis, even stitching. So can we just put into the artificial intelligence and machine learning program, and then they can actually automatically give us recommendations or output for us. So that's what we have been doing. So one more time, we have available technologies, aerospace technologies, and we have a satellite and drones. We have a geospatial technology, we have a GIS and image analysis, and then now we have artificial intelligence, which includes machine learning and deep learning. So putting those together, I think we can detect insects and insect-associated damages in the crop or trees very efficiently. So these are my funding agencies, and we have a lot of transdisciplinary teams to do this project. Thank you.
Thank you so much, Dr. Park. It's always interesting to see the new applications for drones and all the wonderful technology that they can bring for us. I know that within the confines of USDA, they've been using a lot more drones at the port for inspections. So flying them around cargo that would be very dangerous for a person to climb around on, and using drones with that application. So that's pretty interesting. Does anyone have follow-up questions for Dr. Park? We got a couple minutes.
Hi, Dr. Park.
Yeah, person.
Are there seasonal limitations? I mean, obviously the plants are growing at certain times, but I mean, the whole thing of like healthy plants refracting their correct light and wavelength, could you only use the drones to spot problems like within like a certain week of a season, or is it for the whole season you can use it?
Right, good question. So this is what we are doing actually. So let's say the emerald ash borer, right? And they attack ash. When would be the best time to fly? Right? We fly during the winter first, so that we can eliminate all the conifers. Make sense?
Yeah.
And then we fly multiple times to see the progress of plants and things, right?
Because
right.
I guess when it doesn't really cost anything to fly it because you're charging them with batteries. So the initial cost of the drone is really, that's it. And then like the limiting factor would be like professionals' time to go out and do it. Okay.
Right. So depending on what we are looking for, actually it changes altitude, flight altitude, and then time of the day, and then what kind of environmental condition we need. Right. So a lot of people says, oh yeah, sunny day is good. No, sunny day is not good. Actually, the fully cloud day is the best. Right, because of the reflectant.
And just, did you meet with Image? Did that go?
That's really neat. I just wanted to ask real quick. I wonder if you could do something like this, not using thermal reflectance necessary, but I'm thinking of things like adelgids or mealybugs that have a waxy layer. Those guys are smaller. They won't have big flight muscles, but they do have this really reflective, waxy surface that show up as little white spots. And I wonder if that would be a potential application as well.
Right, right. So, insect, like we called insect signs of insects, right? Their product or, skins or, or some plant has a different colors on the upper side and lower side, right? And then actually especially for Hemlock woolly allergy, this study has been done with the drone. So they say they can detect, I cannot say detect, they have a higher probability to say this hemlock tree have hemlock woolly allergies.
Is that from you? Is that from your lab or another group?
No, not my group.
Okay.
Based on NDVI.
Okay, cool.
Yeah.
Awesome. Thank you.
Oh, that makes sense.
Yeah, but the problem is we actually did the same thing and we couldn't detect it. We did a validation study and we didn't detect it. That's why AI is important. So put all the information together and then build up, and that can say something, right? Not just the individual, because the environment is different, weather is different, as Twitter says, right? And then light is different. So, right, it's challenging. So that's why we are moving toward the more like a machine learning with a lot of data.
So when you're looking for emerald ash borer, are you looking for their heat profile underneath the bark as larvae, or are you looking for them as adults when they're feeding on the foliage?
So two things. We try to detect the like a senescing ash trees because they have a, what is that called, a bi-branch pattern. So we can detect actually the ash trees using drone and then image analysis. And the other thing what we are doing is they have an exit hole, right? So they come out from the stem. So we are developing actually drones flying under the canopy.
To look for exit holes?
Yeah, and then the exit hole, this is the thing, the exit hole, right? So if you take a thermal image pictures with the exit hole, the inside is cooler, outside is warmer. So you see like a exit holes actually.
Okay.
So, but again, you know, this is still going on. So we'll see how it goes.
That's cool.
Very good. Thank you. You do have one question in the chat here.
Okay, let's see.
Just curious, is there any identifiable amount of time required for any of these insects to be in flight to be able to be detected by the image analysis?
Okay, so
is it possible for insects to generate detectable heat just before they take off?
Right. So okay, so we did our study just focused on our mason bee because we were making a pollination robot based on our project, our mason bee. So based on actually our study, so the mason bee actually do free flight warming up. So whenever they actually use mussel, we can detect it right away. But if they don't do free flight warming up, we cannot detect it. So warming up means they are not actually flapping wings. They are moving muscles actually to heat it up before they fly. Right.
That's really interesting. So it's the vibration more.
Yeah, vibration is enough to detect.
Gotcha.
Yeah. I really hope actually I could detect 3D crystals.
All right. Well, thank you so much, Dr. Park. We really enjoyed your talk. I'm going to move on in the interest of time here. We have about an hour left of presentations. Again, I didn't really build any breaks into the presentation since it is virtual. I'm just kind of banking on the fact that folks are going to do what they need to do. So unfortunately, our next speaker was going to be Molly Sherlock from West Virginia University, but she is not able to make it with us today. I will try to get her back at another meeting to discuss her ongoing projects. So I think we will just move on to Eric Dodseth. He has been a longtime WVES member, as long as I can remember coming to the meetings. Eric has been there, and he's with DHHR, and he's going to talk to us about vector-borne disease activity in West Virginia. Eric, did we lose you? Are you still there?
Maybe he went to go get lunch because he thought he was after Molly.
No. Oh, no. Okay, well. we'll
Everybody
just
go get lunch.
Let's see, is Dr. Rowan with us?
Yeah, I'm here.
Okay, would you mind, is it okay if we jumble up the schedule a little bit and let you
This
go
is
next?
Eric, actually.
Oh, Eric's back, okay.
I tried using review slideshow from the beginning, and somehow, let's try this. And
Yeah, we can hear you really well. but
yeah. Let me start here. I was trying one way and it didn't quite work out the way I wanted.
Okay, sure.
Okay. Share. And it's loading and doing something.
There we go.
Okay, you are seeing vector-borne disease activity in West Virginia?
It's
Yes.
not in slideshow mode. It's
Right,
Just,
we
it's
just
just
need to choose it.
Okay, I'm going in slideshow. Now it is. It should
Yep.
be, right? Okay.
But we're seeing it.
Everything
it's
good.
like the do you
Like
have double
the preview
monitors?
one.
Yeah.
Oh, really? It's not
So it's showing the both slides, like your
showing
next
both
slide.
slides. I thought I could. I thought I had it from slideshow that would show just the slides. Okay.
So this happens to me when I have another monitor attached.
Yeah, I have two monitors. That's why.
I have to just detach the other monitor.
Oh, I would prefer not to detach the other monitor. Will this cause any problem if people see two slides?
No, unless you have like a funny joke, you don't want it to be real.
I like that. Let's see. We're gonna try this way.
It'll work that way.
I think it'll be fine.
Yeah,
Okay.
I think if
It'll take up
everyone
time.
I think if everyone maximizes their screen, it should still be big enough.
Yeah, it's
Okay.
big enough.
Okay. Okay.
All right.
Okay. I guess.
Go ahead, Eric. Thank you.
Let me see. Well, hello, I'm Eric Dutzett, the State Public Health Entomologist in West Virginia, and I'm going to be talking about vector and vector-borne disease activity in the state in 2020 and 2021. And I'd like to thank, first of all, everyone for inviting me to this. This is a nice diversion from doing COVID duties, so thank you very much for allowing me to give this presentation. I'll be talking again about mosquito and mosquito-borne disease activity within the state, and also talking about tick and tick-borne disease activity across the state, and then comparing our human incidence of Lyme disease in comparison to the black-legged tick, Isodes scapularis activity, using a lot of different surveillance methods that we have available and are starting to use more and more often. And then discuss the emergence of alpha-gal syndrome, which is better known as red meat allergy, and coincide that with the emergence of Lone Star tick within our state. When we talk about mosquito-borne disease activity, for 2020, we had 7 La Crosse encephalitis human cases, which is higher than what we've seen in a few years, but it's still lower than what we used to see in the 1990s and 2000s. So I'm thankful for that, but still it was, I consider, still too high. For 2021, there were no confirmed or probable human cases of mosquito-borne disease in West Virginia, but we still are investigating one of them, so keep eyes on that. For mosquito surveillance, we conducted mosquito surveillance at 18 locations in 7 counties across the state, including state surveillance, as well as from Cabell, Huntington, Monongalia County Health Department, and Kanawha Charleston Health Department also assisted in the mosquito surveillance activities for 2021. This year, we could not conduct mosquito testing at our state lab at OLS, which I like a lot. They've traditionally been able to test our mosquitoes for La Crosse, West Nile, Eastern Equine, and St. Louis encephalitis. This year, they were so swamped with COVID responsibilities, we had to send these off to CDC for testing, and it had to be very limited testing, unfortunately. But we're still able to do some mosquito surveillance for this year in 2021. We did detect West Nile infection in one of our mosquito pools in Cabell, in an area where we have seen West Nile virus activity in humans as well as mosquitoes in this area. And in our site in Kanawha County, where we have seen West Nile virus infection within the mosquitoes within that area previously in previous years. Now to shift to tick-borne diseases, here's again a listing of the tick-borne diseases we have, the pathogens responsible for them, and the tick vectors that we have. As some of you have seen, this has been a historical, but we have been adding more pathogens as well as more tick-borne vectors that we find in West Virginia. And some of these you can question, but they still could be, including tularemia. I think of that more as more of a wildlife situation, but taxidermy situation, but it could be transmitted by ticks. For tick-borne diseases in 2020, again, our #1 disease was Lyme disease with 1,062. It's been increasing each year, but we have also had spotted fever, rickettsiosis, included Rocky Mountain spotted fever, ehrlichiosis, anaplasmosis as well. For 2021, again, we are still reviewing the cases, so this is incomplete. This is not a misprint. We had 1,063 Lyme disease cases ascertained as confirmed and probable as of December 29th, 2021, but we have a lot more left to investigate. The amazing one has been ehrlichiosis. We've seen a dramatic increase in ehrlichiosis to 14 cases. This has been predominantly in the Charleston, Huntington area, where we're seeing a lot of Lone Star tick as well. We also have been seeing anaplasmosis as well as spotted fever, rickettsiosis as well, and one tularemia case. As I mentioned before, we have been seeing a lot of Lyme disease. It's been increasing dramatically following the emergence and spreading of black-legged ticks, which I mentioned in previous presentations. And it looks like it's going to again increase for 2021. Here's an old map, but it gives a nice visual showing again where we see a lot of our human cases of Lyme disease. And I use this one so I can show you this other map showing where we're seeing our high human incidence of Lyme disease. These are human cases. Again, the darker areas showing where we're seeing most of our Lyme disease cases being the eastern part. This will influence what I'm going to be later discussing about our surveillance plan for tick surveillance. This is showing human incidence as a function of number of human cases for every 100,000 people. Again, we're seeing it a lot in our eastern part of the state is where we're seeing a lot of our human cases of Lyme disease. Now, a few years ago, CDC developed a protocol that should distribute to state health departments discussing surveillance protocols, standardizing surveillance protocols for tick surveillance around the country. And in the past, we have done very well. I would say we got a pretty good idea of the ticks that we have here, their distribution and seasonal phenology for a lot of them, including the black-legged tick, Isodes scapularis, vector for Lyme disease. But we still have a long way, and this was through our work through West Virginia Veterinary Tick Submission Project, through our collaboration with the DNR, public submissions, as well as from hospital submissions, as well as limited active tick surveillance. But this is not going to be very helpful for other things that we really want to do, including looking at the infection rate with the black-legged tick Isodes scapularis for Lyme disease. We want to look at the densities of those ticks, as well as the infection rate for Lyme disease within those tick populations, and looking at that as a function of density of those ticks. Those are the next projects that we've been doing for the past 2 years, and this is going to involve more active surveillance on our part, as well as limited with our veterinary tick submission projects, as well as public submissions. Again, we are interested in the black-legged tick Isodes scapularis. That is a tick vector that's most responsible for Lyme disease within the continental United States, but I should say the eastern part of the United States. And we do know that black-legged tick Isodes scapularis are infected with Lyme disease bacteria in West Virginia. We've seen that time and time again. Looking at transmission cycles, this is a review. The larval ticks are not the ones that do have the infection, although we do know that it is transalveolally transmitted, although very limited from the mother to her offspring. Predominantly, these larval ticks are acquiring the Lyme disease infection from infected white-footed mice, which are shown here in green. That's not the way they are in nature, but it's just for visualization. And the larval ticks will feed on these white-footed mice, pick it up, the nymphs will become infected, contain it for the rest of their life. They will then start feeding on another host, which could include other field mice or other medium-sized mammals, including humans for that matter. Then from there, the adults will have infection and still continue to hold infection. And then they will start feeding on other larger mammals, including deer, as well as humans. So again, when we're concerned about disease transmission, we're looking at that nymphal stage as the one that's acquiring it, that first initial acquiring of the Lyme disease spirochete. Most of our human cases of Lyme disease tend to be in June and July. This coincides very much with when we see the nymphal tick stage for Isodes scapularis, the black-legged tick. That's when it's most active. So we have very good reason to believe that the main stage is transmitting Lyme disease to humans is going to be the nymphal stage. And because of this is influencing when we're doing and how we're doing our surveillance plan. Our active tick surveillance activities are going to be focusing on, again, the black-legged tick, Isodes scapularis, Lone Star tick, because it does transmit some diseases and it is emerging, and Asian longhorn tick because of the fact that it's an invasive species. that is also capable of transmitting, as we found out recently, Rocky Mountain spotted fever, and we're hearing more and more situations of humans getting bitten by this tick. It may be a situation of this invasive species is having a host shift, or we don't know yet. Time will tell. Our active tick surveillance was again for 2020 conducted from April 5th to July 21st. This is when we're looking for the nymphs for Izoia scapularis. We switch off. We aren't interested in doing much tick surveillance from, I would say, early summer to fall. That's when we get the larvae for Izoia scapularis. I'm not as interested in those, but we still want to keep an eye on those. We then conducted more active tick surveillance from September 30th to December 8th for the adult Izoia scapularis, and this was in 28 localities and 18 counties that are shown in green. As I said before, we did limited tick surveillance from July 21st to September 30th for the larvae. We use a standardized method. We're looking at a minimum of 1,000 meters squared, but we also do receive passive surveillance from our veterinary colleagues, as well as from public submissions as well. Here again, this is just a quick listing of localities, populated localities near those, the county, and then the number of tick nymphs, Izoia scapularis tick nymphs we get per meter squared, as well as just for every 1,000 meters squared. Here's just a box whiskers plot. Again, I don't want to use any statistical analysis on this. Again, we were interested predominantly in just finding infection rates and just generalized densities at 1,000 meters squared. I apologize for this. But what we found were black-legged tick Izoia scapularis were active in every locality that we looked at in 2020, except for a locality in Huntington, West Virginia, where we don't get much Lyme disease in Cabell County. So this supports pretty much what we're seeing. Most of the collecting localities with our highest black-legged tick Izoia scapularis nymph densities were in the eastern part of the state, and this is where we're getting most of our Lyme disease, so it shouldn't surprise us too much. And surprisingly, a lot of the sites where we're seeing a lot of our Izoia scapularis high densities tend to be closer to humans, and we suspected this before, and we've heard this from our colleagues. We suspected this early on when we were looking at our deer, and we're looking at the number of deer and how many ticks that were infecting those black-legged ticks. And we're finding a lot of the deer that had high infestations of black-legged tick tended to be closer to human habitat, or where we're paradomestic habitats. And this is what we're hearing from other of our colleagues as well. We've submitted our black-legged tick Izoia scapularis ticks to the CDC for human pathogen testing. We're interested in Borrelia burgdorferi and Borrelia mayoni. These are the two pathogens responsible for Lyme disease. Borrelia mayoni more in the Midwest, but we were just curious if it could be down in West Virginia as well. Borrelia miyamotoi we were interested in. This is relapsing fever. We haven't seen relapsing fever in our state. However, we have been seeing it in neighboring states, so we are very concerned about it being in here. Anaplasma phagocytophilum and Babesia microti, because it is becoming a problem here in our state. So these are some of the pathogens we were looking at. So far, we found 16% of the Izoia scapularis nymphs from Kanawha County were infected with Borrelia burgdorferi. This was amazing. I've been surveying at this site and testing at this one site for years and have not seen any infection with Lyme disease spirochete, but just this past year we found it. So I was very excited about that. Horrified too, but it is showing that we are seeing an increase in Lyme disease infection within our ticks, and we also saw an dramatic increase in Lyme disease cases in Kanawha County. 2% of our black-legged tick nymphs from Kanawha County were infected with Anaplasma phagocytophilum, and 13% of our black-legged tick nymphs from Greenbrier County, which is in that southeastern part of the state, were infected with the Lyme disease spirochete, and 15% of those nymphs were infected with Anaplasma phagocytophilum. This is relatively high for Anaplasma phagocytophilum, considering the fact we don't get many human cases of this in the state. However, this has not been typed. There are different typings of Anaplasma phagocytophilum, and some of them are a little bit more virulent to humans and more pathogenic to humans. So this could be the more deer variant type. As for the infection rate that we saw with Lyme disease within our black-legged ticks in this part of the state, we thought this was relatively low, but some of our colleagues in Virginia were also reporting about 13% infection rate in the, I would say, the western part of Virginia. So it is collaborating with what we're seeing with other, from other tick surveillance activities. We've also submitted some of our ticks from the 2019 West Virginia Veterinary Tick Submission Project to the new West Virginia University Vector-Borne Disease Infectious Disease Laboratory. And we wanted to test them for Borrelia burgdorferi as well as some Rickettsia symbionts, Rickettsia buchneri. And we're curious to see if Rickettsia buchneri may be somehow enhancing transmissibility of Lyme disease. Maybe it's making the tick more survivable. Maybe it's modifying its behavior. Well, time will tell, as Timothy Driscoll, who is heading the Vector-Borne Infectious Disease Laboratory at WVU, is looking at this a little bit more closely. And we hope that he is able to get on more, do some more with this. Right now, I hear he's quite swamped with COVID-19 responsibilities like the rest of us, dealing with wastewater and COVID-19. Lyme disease infection, we wanted to also see Lyme disease infection in our companion animals and other surveillance methods, including our companion animals. Now, IDEXX developed the 4DX SNAP test to test for a lot of pathogens, including heartworm, as well as antibodies for Lyme disease or ehrlichiosis and anaplasmosis within dogs. And this information is available to the Pet Health Network. It is limited. The counties that are reporting are counties where the veterinarian is, not necessarily where the animal patient is. And furthermore, this SNAP test is based upon antibodies that could have been in the animal for a very long period of time. Shows past infection, not necessarily recent infection. So there are limitations to this. However, we have been seeing an increase in Lyme disease infection, at least it looks like it's encroaching in the western part of West Virginia, where we have so far had low human cases of Lyme disease. We also use syndromic surveillance. This is a method that has been recently used within health departments to look for timely system for detecting health events. A prominent one that we're seeing is like for influenza, for example. How many times are people going to the emergency or urgent care to look for influenza or upper respiratory infections without necessarily going through the full gambit of the laboratory diagnostics? It's a way for us to monitor very quickly disease activity. So we were interested in looking at how many times people were going in for tick bites, for example, and for tick encounters for urgent care or emergency. And we did see a dramatic increase, I guess, in tick encounters and tick submissions to urgent care and emergency room visits. And this is shown by darker areas, darker counties, showing having more visitations from people for tick exposure. But again, as we all realize, there's a lot of tick species. There are a lot of tick species that will bite humans. So we had to somehow differentiate, try to partition to see if it is truly black-legged tick, realizing that black-legged tick, Azoia scapularis, is really the only tick that we tend to see being active in the fall, being October, November, and December. Those are the ones that are actively biting people during that time. So we wanted to see if we're seeing a lot of tick encounters around the state, particularly in October, November, and December. And it seems to be the black-legged tick is becoming more active in Western West Virginia. And this is what we're seeing for, again, for the fall months, being in October, and again in November, and again in December. So what we're seeing is a situation where we are seeing, it looks like more, at least animal cases of Lyme disease in the western part of the state. We're also seeing what look like possibly more black-legged tick nymph activity, or black-legged ticks, I should say, in the western part of the state. But we really weren't seeing that many human cases. So we wanted to see what was going on. So we decided to look at the tick profile there. So again, a lot of tick surveillance for 2021, looking at a lot of the same ticks that we saw previously. And we did active tick surveillance in January 18th to August 4th. Yes, I do tick surveillance, active tick surveillance in the winter. Obviously not today for a lot of different reasons. But when it's a little bit clearer and we don't have as much snow cover, they are definitely out there. I look ridiculous at times, but it's something that they are active. Black-legged ticks are still active during these winter months. And we had limited tick surveillance conducted from August 4th to December 20th, because that's when we were focusing a lot on our mosquito surveillance activities. And then again, we did pick up tick surveillance again after September 20th. Ticks were collected using a minimum of 750 meters square. We went down a little bit, but we still had our passive tick surveillance from our veterinary colleagues as well as public submissions. This is again looking at the localities and counties that they are and the densities. Again, our box whisker plots. And what we found were a lot of our black-legged tick, Azoia scapularis, were not active in many of our collecting localities in southwestern West Virginia. So therefore, what we're seeing is that we just don't have as many black-legged tick nymphs in the western part of that state, that southwestern part where we're not having human cases. So it appears that the tick profile is very much coinciding what we're seeing with the human aspect of things, and our human Lyme disease surveillance is overall recording what we suspect is a very true statement. We don't have as many hospitals and laboratories in that southwestern part of the state, so we were concerned about this, but it looks like there may be truth. We're just not having many Lyme disease cases in the southwestern part of the state. Most of the collecting localities with the highest black-legged tick nymph densities were in the eastern part. That's where we have a lot of our Lyme disease. And again, many of the sites with high tick densities for Isoia scapularis are near human habitation. And now I'm going to switch gears. Now I'm going to be talking about alpha-gal syndrome, better known as red meat allergy. And I give these presentations to health departments and the public and school groups. And I'll talk about Lone Star tick. And I think they fall asleep when I talk about Lone Star tick. When I talk about, okay, human ehrlichiosis. We suspect it is a vector for spotted fever rickettsiosis. Star eyes is still up in the air about that one. But as soon as I talk about red meat allergies, and this is the cause of it, their ears perk up because I like eating my red meats. I like my barbecues as well. So I'm very sympathetic. This is a cause, it's a hypersensitivity reaction to galactose alpha-1,3 galactase, which is found in non-primate mammalian meat. Not only is this used directly consumption, but we also do have a lot of products that are made by these non-primate mammalian meats, including gelatin, which we all like ice cream. So it's found in ice cream. It's also found in heparin, which is an anticoagulant. So this has definitely medical significance, as well as it's found in cosmetics. I don't want people to be allergic to their cosmetics. Unlike a lot of our food allergies, this is delayed for up to 2 or more hours after exposure following consumption of meat products. The clinical symptom is broad. We don't really have a cluster that we can clearly define associated with alpha-gal syndrome, but this is a situation where patients will develop, it will bind to the antibody. It will cause a releasing of a histamine, which will result in anaphylactic shock, as well as urticaria, which is sort of I would say an inflammation, skin inflammation, a skin reaction that we see. And anaphylaxis, whereby we have a situation where we're having a dilation of our blood vessels, which will cause a decrease in our blood pressure, which will make people feel faint and fainting spells. It'll also cause a constriction within our bronchioles, within our lungs, making it very difficult to breathe. Patients with this also develop gastrointestinal and respiratory symptoms, as mentioned before, and this is following consumption of meat products, non-primate mammalian products. Diagnostics include whereby they will cause an incision on the skin of a lot of these different mammalian meat products and see if we have any allergic reaction to these. Management for these include the use of antihistamines as well as epinephrine and the elimination of meat products from your diet. Unfortunately, this is a situation where once you have it, it takes a very long time to get rid of it. You can decrease the severity over time, but I have not heard of any specifically good treatments for this. The majority, again, of these situations, these patients overlapped with the distribution. This was in the southeastern part of the United States. And it took quite a creative endeavor to find out that actually this overlapped with the distribution of the Lone Star tick, Amblyomma americanum. And they found that there was actually an increase in IgE following the bite of a Lone Star tick. And alpha-gal has only been so far associated with tick bites. So that's interesting to see. And it has now become on the national radar. We have our annual CSTE meeting, which is Council of State and Territorial Epidemiologists, to discuss how we need to modify our case definition for disease monitoring across the country. And one of the ones that was posited was making alpha-gal syndrome a reportable condition. This was actually proposed by Virginia in the 2021 meeting. And now we will see, it will be discussed with our group. I'm proposing that we make it reportable in West Virginia. It'll have to go through our state epidemiologists and our legislature to make it so. The Lone Star tick is characterized, as the name implies, not by the fact it's from Texas, but it has that pearly white dot on the posterior scutum of the female Lone Star tick. This is a tick that, unlike our black-legged tick, which these will be found in deciduous forests, but we tend to find it a lot more in undergrowth and high grasses. near secondary growth, and you also find these in lawns as well. So this is one that will venture a little bit further into the lawn environment. As an aggressive generalist feeder, I have been chased by these things. This is a sexually dimorphic species. As I mentioned before, the adult female will have that white spot. If you see a tick that has an anal groove on it, it is going to be Isotes species. But if you see a tick that is a little bit more circular in dimensions, it has festoons along the posterior, and if it has long mouth parts, this is going to be belonging to the genus Amblyomma. And there are 2 species that we have of Amblyomma within the state, including the Lone Star tick, Amblyomma americanum. We also do have Gulf Coast tick, Amblyomma maculatum, which so far has very limited distribution. We have 1 population in Mason County, but we do see it sporadically occurring in the state. So we need to keep an eye on that. The adult Lone Star tick is differentiated from the Gulf Coast tick by having a less elongate internal spur. If you were to look at the nymphs, the nymph of this tends to have, of the Lone Star tick, tends to have a more rounded basal part of the mouth part, as well as the first coxal segment having 2 spurs instead of 1. Here's just a general distribution of the Lone Star tick, but if we were to look at the state, there's the blue show where we have spotted it. The red is showing where we have established populations. So we see a lot in the Charleston, Huntington area. This is where we see a lot of our Lone Star tick. And this is where we're also seeing a lot of our ehrlichiosis cases as well, which is vectored by this tick species. This is a tick species that feeds on 3 hosts. Each stage will feed on a different vertebrate host. The adults and the nymphs are most common late spring through the middle of summer, whereas the larvae, which are most common in late summer. And when I hear of seed ticks, and when I hear of people getting infected with hundreds, if not thousands of ticks, this is the tick that I think of. And I have been a victim of this, so I'm very sympathetic. The majority of our ehrlichiosis cases we tend to see in the summer months, typically in June and July. This coincides with when we see the adult and nymphal stages of the Lone Star tick. And all stages of this tick will feed on humans. I am a victim of this. However, only adult and nymphal stages are the ones that will carry Ehrlichia chaffiensis. For management, for personal protection, we always encourage personal protection. This will tend to be one where we tend to see a lot of tight fitting areas, barriers, if you will, all the way with the back of the knee to around the belt area, armpit, groin area. Again, I encourage people to use tick repellent, but have a higher concentration, checking the body after you're outdoors for activities. This is one, unfortunately, you'll get, particularly in the later part of the summer, where you will get hundreds of these things. So it's going to be a little bit more difficult for personal protection, but again, we encourage that. For management, this is a new area. Controlled burns can work. It can work on an annual reduction, but again, once we get the new mammals coming in the following spring and summer, it's going to come back, unfortunately. For deer removal and for deer fencing, I'm not as optimistic as I would like to be on using these techniques. For acaricides, we used to have a lot of organophosphates that were really quite effective, but we really can't use a lot of those organophosphates down residential areas where we're going to see a lot of these human cases. Pyrethroids can be effective, but again, I can't guarantee on the efficacy. It's definitely not going to be available. It's not going to have long-term. It might last for about a month, month and a half or so. Natural acaricides can be used, but again, we don't know how long or how effective this will be. Fungal acaricides, as I was mentioning with the previous presenter, this is a new area, so we are looking for fungal acaricides. Possibly this will be the new ticket. Bait boxes, I'm not going to be as gung-ho about this. It's been looking very well for black-legged tick Isotes scapularis because of the fact that this will target the larvae. As these rodents bring this material to their den, it will be attacking those feeding larvae. But unfortunately, the larvae for the Lone Star tick does not feed on rodents. So in summary, the number of lacrosse encephalitis cases was low in West Virginia, comparatively low if you were to look back in the 1990s and 2000s, but it was high in comparison to what we've seen in the past few years. The human cases of Lyme disease, as always, are seen to be increasing. The density of black-legged tick nymphs is higher in our high incidence Lyme disease counties, but lower in that western part, the southwestern part, where we do not see a lot of our Lyme disease cases. And alpha-gal syndrome is caused by the Lone Star tick. And that's my contact information. If anybody has questions, let me know. Thank you. Appreciate it.
Thank you, Eric. That's really interesting stuff. And I had not heard of the alpha-gal syndrome being related to the Lone Star tick.
And
So
I hope nobody has to. But,
yeah,
we have been actually, I want to follow up on this. This has become a concern because we are hearing school cafeterias having to modify their diets and what foods they
Oh.
provide because of this. And some parent groups are saying this is ridiculous. Even some of the cafeteria people are saying, why are we having to prepare this? We don't think this is a real thing. So we want to provide information and show, yes, this is a very real concern. We want to see where this is approaching.
Sure.
Yeah.
You do have a few questions in the chat there if
Okay,
you want to
yes,
address
yes,
those.
yes. Let's look at these. Let's see if I can. Fortunately, let's see. Fortunately, I minimize it, but I will. Let's see here. Okay, here we go. Chat. Okay. Okay, how many of the actual cases of lacrosse, encephalitis, or West Nile fever get recognized or reported? I think that these, as well as Lyme disease, are greatly underreported. I would agree very much with you on definitely with a lot of these. Lacrosse, encephalitis, this was a big thing in the 1990s, 2000, definitely. And a lot of our sanitarians and our public health nurses that used to deal with this are retiring. Yes, you are correct with that. And a lot, as well as physicians and laboratories. A lot of those used to be dealing with this. I suspect this is underreported, and I get very concerned with COVID-19 and the responsibilities for a lot of these hospitals are saying, we just don't have the time to thoroughly investigate this.
Let me add to that real quickly. If you have a disease and you don't look for it, it does not occur there. If it does not occur there, why waste my time looking for it? And that's part of the real problem. If you look for it, you will find it.
Yes, yes. However, we have been seeing with our lacrosse cases, these are in areas where we've historically had lacrosse encephalitis. But then again, that answers what you're saying. Well, if they're looking for it and they've had it there for the longest time, then they're going to keep looking for it. That is true, but this is a requirement. This is a reportable condition. Laboratories are required to report this across the state.
If they look for it, and the problem is if you're an area where lacrosse is not known to occur, then why waste my time looking for it? Because you don't find it, you don't look for it. If you've had it there in the past, you may look for it. It's an unbelievable problem with underreporting.
Yeah, this is why we do use a lot of the other reporting. This is why I'm very much a supporter of our One Health approach, looking through mosquito and tick surveillance as well. In the past, we have done a lot with, I like to brag, about our OLS testing capacity for mosquitoes. And we have gone in large areas, including that northern part of the state, Monongalia, as well as the western part of the state, and testing those for lacrosse encephalitis. But you are correct in that regard, it is an underreported disease. But I was hoping that with a lot of our additional surveillance techniques, we can detect that. And we did have a situation a few years ago where we did see lacrosse in some areas we didn't notice it before, and it has not been traditionally reported, and that would have been around the Cabell area. And it was interesting when we saw an increase there, we actually saw an increase of lacrosse encephalitis across the river in Ohio. But thank you for that reporting. Lyme disease, yes, that is one. A lot of our physicians, the good news is a lot of them do know about it and are treating them with doxy, but is a situation where they just treat it immediately. They're not going through the diagnostics. It's just, we'll just treat them, very much so, which I am thankful they're treating at least the patients. Thank you. Can the Isodia scapularis become infected if feeding on an infected deer? I thought that mice were the source of infection for ticks, but deer were critical for tick survival. The deer story. So far, we have no indication that deer would be actively transmitting to ticks. So far, it does not look like they are the reservoir for this. And I haven't seen anything yet that would suggest that deer would be the reservoir for this. Unless, Michael, have you heard of anything new? I have not seen anything. I have not seen them being able to carry it. It almost seems like they're refractory to it, if you will.
I mean, this is the same, it's true for some of the California encephalitis viruses too, like lacrosse, Jamestown Canyon. Deer become infected, but they don't produce a viremia. So although there's a high infection rate in the deer do not seem to be a source of infection for the ticks or the mosquitoes, depending on the disease.
Yeah. How about Jamestown Canyon? You don't think they're developing a high enough viremia? Well, they have to be developing a high enough viremia for Jamestown Canyon.
Why?
Why? I always thought Jamestown Canyon was transmitted through deer, and that was the main source, unless you see something different. For those who are unfamiliar, Michael Terrell is the prominent, I would say, one of the prominent experts on medical entomology. So insights that he provide are great and wonderful. So what do you think, Michael?
Personally,
Dr.
I think
Terrell.
Mike, I go by
Okay.
Mike like a bug man. But
Okay.
for vector-borne disease, particularly the California group, vertical transmission, transviral transmission can be extremely efficient in a subset of the population. Essentially, 99%, 100% of the progeny are infected. So the virus can persist forever without any vertebrate involvement. We looked at, for some of the California viruses in California, looked at infection rates in the cattle and other animals, in the mosquitoes. And if you saw an amplification, you would expect to see higher infection rates over the summer. It was flat. Therefore, the mosquitoes are infected vertically, but are not infected by feeding on the infected mammal, or very few of them become infected feeding on an infected mammal. The virus is maintained by vertical transmission rather than horizontal. The California group is just maintained. It's a mosquito virus that happened to adapt to feeding to growing invertebrates and causing disease in some of them, i.e. lacrosse encephalitis in kids.
Yes, yes. And excellent. Thank you. Thank you for
Thank
those
you
insights.
for your talk. It was very good.
Okay, we have another one from Ed. Ed is also one of our medical entomology people. Thank you, Ed, for participating and always be a member of West Virginia Eds. He's also one of our great cave explorers. I've collected Azoia species, likely Scapularis, in every month of the year on our property in Monroe County. Monroe County is getting swamped, Ed. Yes, it's very much, and we actually do have samples that we're sending off to CDC. So thank you for your insights on that, and always contributing the specimens that you have over the years.
Yeah, I just wish we'd find fewer of them.
Next one, could you tell us more about the new disease vector lab at WVU? Willing to take public samples? What all the testing are planned to test? That's going to be one with Dr. Timothy Driscoll. He started that up a few years ago where he officially changed the name to that. Right now, he's been doing a lot with COVID-19, swamp with those duties. I've had a hard time at times getting in contact with him. He's looking at wastewater and COVID-19 infection within the wastewater, really exciting stuff. As well as I think they're going to look at some other diseases as well. But I believe they will take specimens. I know they've been talking about they have taken some from us as well. They're looking at human pathogens as well as rickettsial into symbionts. And we hope that they're able to contribute a lot more in those endeavors. Okay, next one from Jennifer. I've heard that alpha-gal is responsible for the incompatibility of non-primate organs with humans that need the organs, and that recently they genetically modified pigs. Well, that's new. And that 2 pig kidneys. Oh, thank you for that. Thank you for that. I appreciate that. And then Yong-Lak, any update on triatome in West Virginia? Yes, we do have Triatoma sanguisuga, and the ones that we did get from Doddridge County were positive. Oh, yes, it was positive from there. Okay, thank you again, Yong-Lak. He's been working with our program. He was actually the first one to actually see the Triatoma sanguisuga, actually. And he contacted our lab and said, Eric, help. Please send this off for testing. And we did test it. We did contact the patient. Tell them, please contact your physician. Tell them that here's my letter saying we did see it within the kissing bug. Please get tested for this. I have not received any lab reports about this situation, so I don't think he was tested on that for that, unfortunately, and I have not heard anything yet. But we do, and we have seen infection. We have seen infection of kissing bug as far north. I know we've had some around the Marion County area as well. So it is in this area. I could pull out, we have been seeing a lot more of that in, I would say, the southwestern part of the state, where we have seen a little bit higher infection, a lot more of those kissing bugs being infected with Chagas disease, and have been feeding on humans as well. That's another thing. there has been human blood meals from that. And I know that Yong-Lak is also interested in looking at the biology of kissing bugs within the state as well. So any other questions anyone has?
Can I ask a really simple question about
There's no
the
such
life
thing.
I guess so. So I've had ticks on like my dogs and stuff like that growing up. And it seems like there was like always, I mean, once a tick was on it, I assumed that they were just on it forever. But it seems like the tick in the wild as a nymph will go on a mouse and then will it go off in order to go on a deer?
Yes.
Why does it do that? I never thought that they lived long enough to be like, okay, I'm done with this food source. I'm going to go waste energy to go find a harder moving food source.
Let me just make a quick comment on that. That ticks, you have one host ticks. That tick will be, like you said, get on it and stay there forever. Those are very rare. Most ticks, the larval stage will get on a small mammal, take a blood meal, drop off, and molt into a nymph. That nymph will then find another vertebrate host, take a blood meal, and drop off. Most animals do not like having ticks on them. If they find the tick on it, and it will kill or disrupt the tick. Therefore, once you get your blood meal, go away in safety, molt to the next stage, do it again. If a mosquito were just to sit on your arm forever, take blood whenever it wanted, it would die. Same problem for the tick. It feeds only as long as it has to be there to get blood, which may be up to 2 weeks for an adult to get a full blood meal.
Excellent explanation. Thank you. I was also going to comment that the ticks also, some of these are less desiccant hardy. They really need to get off there. I've had black-legged ticks I've collected from the field, and I would accidentally leave them on my dashboard for like 5 seconds. They're dead. Oh, great. Okay. So I would see also just being on that animal for that long period of time in a less humid environment for some of these ticks is also kind of dangerous for them too. So thank you.
Do they have to go to a deer? So they go on a mouse first and then they have to go on a deer?
Different ticks, each tick has its own set of animals that it's adapted to grow on. The deer tick or the black-legged deer tick, the adults tend to go to deer. So if you don't have deer, most of the adults don't get a good blood meal. I mean, some of them will feed on a person. They don't all feed on deer, but the vast majority do. A lot of other ticks will never feed on a deer. So it depends on what species of tick you're talking about.
Okay, cool. Thank you.
I was going to comment too about tick feeding and all that, the concept of specialists and generalists as we were addressing and the problems associated with trying to figure out, is this a specialist, is it a generalist? We always find new and exciting things. So.
All right. Well, thank you so much, Eric. Thanks for all the follow-up questions. Those were great.
Karen has one.
Oh, I'm sorry.
Go ahead, Karen.
No, it's just a comment. I just wanted to mention, if you're interested, there are 2 Radiolab episodes that talk about the alpha-gal situation. And the second one talks about the pig genetic modification transplant thing that
That's really cool.
got there. So the first one is kind of the original story of someone who had a meat, an unidentified meat allergy and how that played out. And then the second one is a follow-up to that. And they're both very interesting episodes. Radiolab.
Is that a podcast?
Yes, it's a podcast. So you can get it on
Dating
iTunes
myself.
or Spotify or anywhere where you look for podcasts.
Sometimes it's really good. And sometimes you're like, please stop describing what a thermocycler is. Just say it's a thermocycler.
Yes.
Okay, awesome. Thank you. Well, in the interest of time, we are going to move on. But I really appreciate your time. Eric, you have so many responsibilities, and now with COVID, I know it just seems to never end. So we really appreciate you still taking the time to come talk with us.
Yeah, I'm concerned about WVU coming into admission soon, and I'm kind of nervous about that.
Yeah.
And
I know that I've emailed Eric numerous times, like if I have someone who's really freaking out about a tick, he's always the first person that comes to mind. So he serves a really important role at DHHR, and we're happy to have you as part of the group. So moving on, last but certainly not least, and I appreciate her patience, is Dr. Elizabeth Rowan. Sorry, Dr. Rowan, when did you start your position with WVU?
August 2020.
Okay, I didn't think it had been very long. So, and in the midst of COVID. So unfortunately, I think I've only gotten to meet you one time. You were at Watoga, correct, at the summer meeting?
Some of it, yeah.
Some of it, yes. We had some communication issues down at Watoga. But she is a member of West Virginia University, and you have extension responsibilities too. Is that correct?
Yes, yes, I do.
So she is going to be presenting today on "Do Insecticides Break the Promise of Conservation Agriculture Using Cover Crops?" And then just whoever's left on the call, please know that after this, if you would like to, we are going to have a business meeting, which will probably go an hour or two. But
And be really fun and interesting.
yeah, it'll be amazing. As our business meetings always
It's not
are.
boring. It's going to be great.
So please welcome Dr. Rowan. Thank you.
Okay, hold on. I'm using this computer for the first time for Zoom, and it's going to make me quit Zoom and restart. So I'll be right back.
Your haircut's really cool, by the way.
Oh, thank you.
An interlude. Yes, I have gotten a lot more familiar with extension agents in the county after spotted lanternfly. I have to say that we had a limited contact with extension for the most part, but over the past 2 years, we've gotten very familiar with a lot of our extension agents. So
that's
it's
cool.
nice to have them on board.
While she's loading, oh, she's back. I would love to know more about what's going on with SLF in the Panhandle later.
Oh, sure.
Okay.
It's not good news, I can tell you that.
I have a feeling I know what happened.
Okay, Elizabeth, I have to give you a co-host. Hold on.
Yes, thank you.
Hold on. It works?
Yes.
Okay, good.
Great. Thank you. Sorry about that. I getting to do all kinds of new things with Google or with my PowerPoint. So for example, I have closed caption on, which I'm going to try. So it's been so fun to listen to all of these really diverse talks this morning, and I'm going to talk about something completely different also. So I'm talking this morning about the interaction between insecticide use in field crops, corn and soy, and cover crops. So this is work that I did at Penn State. And it was a fieldwork partnership between myself and Kirsten Pearsons. And then we had other projects going on at Cornell. There were other projects that are associated with this at Cornell in Kyle Wicking's lab and at the University of New Hampshire in Rich Smith's lab. So just to give you sort of the background here, global soils are degrading as we increase the intensity of agriculture. And one of the ways to combat this is to adopt conservation agricultural practices, including practices that reduce tillage, so reduce the amount of soil turnover, increase the diversity of crops that we're planting, and protect the soil with cover crops or mulch. And the adoption of these conservation practices is growing globally. In the US, cover crop use in particular is growing in the Mid-Atlantic region, partially to help retain nutrients, reduce soil erosion, and protect local waterways, particularly the Chesapeake Bay. These cover crops also are thought to improve pest regulation. And the reason that cover crops may help with invertebrate pest regulation is that they tend to increase predator populations. Planting cover crops increases the microhabitats available to predators, which reduces predation by other predators. So it reduces intragill predation. It also can foster the community of small soil prey that those predators rely on in sort of non-pest times. So things like mites and columella are really important prey sources for most, especially ground-dwelling predators. And finally, it extends the sort of the cover and resources throughout the year. So they have sort of better conditions to start their overwintering habitat. So in the fall and the spring, they'll have sort of more cover. However, while we're adopting these conservation agricultural practices, the use of preventative neonicotinoid seed treatments is also growing, especially in the US. So this is just a couple of pictures of what those seed treatments look like. So if you see seeds that have green or red or purple sort of stuff on them, those have a seed treatment. The colors don't really matter. There's not like standardized color system. They'll often be a combination of neonicotinoids and fungicides or fungicides alone. But it's becoming very, very difficult, in fact, to buy corn or soy seeds without a seed treatment on them. So even folks who are looking to buy sort of high quality seeds, especially glyphosate resistant or BT seeds, have a really hard time getting them without the seed treatment on them. And as a result, the use of neonicotinoid seed treatments is there's a huge amount of these products going onto the landscape on millions of acres of farmland across the US. So this map here is from 2014, which was the last year that the USGS was tracking the use of seed treatments separately from other separately. And you can see in areas of the Midwest and in the Mid-Atlantic, we have more than 1 pound of neonic being applied to the soil per square miles. It's quite high levels of neonicotinoids, and this is only clethianidin. There are other neonics that are being used as well. So you can imagine that the application of this much insecticide sort of preventatively on the landscape can be quite a problem for predators. The advantage of using these seed treatments for farmers is not only are they super available, but they are relatively easy to handle. They have very low mammalian toxicity relative to other types of insecticides, and they sort of act as an insurance policy against very difficult to control soil pests and early season pests. So our first question that we wanted to ask was, do these preventative neonicotinoid insecticide seed treatments disrupt those benefits, especially those predator benefits that cover crops are providing to corn and soy systems in Pennsylvania in particular? The alternative to using these preventative seed coats is to use a sort of reactive pest management plan, an IPM pest management plan that's reacting to pest populations rather than sort of an insurance policy. And our expectation is, while IPM might be harder to implement, it is going to reduce the insecticide load on the landscape and hopefully better protect those predator populations and have equal, if not better, pest control. So to test these two questions, we did a 3-year field experiment in a corn soy rotation on 2 fields in at the research farm at Penn State. And we had a replicated experiment with 6 treatments. So control with and without a cover crop, the IPM pest management treatment with and without the cover crop, and the seed coat, which was a neonicotinoid and a fungicide mix with and without a cover crop. So this we're going to call no pest management, integrated pest management, and preventative pest management for later. So the data I'm going to show you today is going to be early season pest populations, particularly white grubs, which are our main pest, plant damage from a variety of pests, including slugs and caterpillars, and then ground predator activity early in the season, predation rates, and yield. And I also want to note that our IPM treatment, we did use a pyrethroid in 2018, which was the second year of the study. And we applied it as a granular in furrow treatment. So as the seeds were planted, we had put a granular pyrethroid in the ground to try to combat the white grub populations that we had measured in 2017. So our IPM treatment had 1 year of treatment, and it was the 2018 treatment. So the other thing I need you to know is that in some cases, instead of looking at cover crop versus no cover crop, we measured total cover early in the season, and that was the cover that was killed, but it included both weeds and this grassy cover crop that we planted. So sort of gives us a better idea of what the predators and pests were really experiencing in terms of ground cover, as opposed to what we sort of put in there, right? So the cover crop and the weeds probably don't function that differently from a predator's point of view, especially once they're dead. So the first data I want to show you is the relationship between cover, particularly total cover, and white grub populations. So we had measured the total cover biomass early in the season that then died. I'm going to show you this is a standardized total cover biomass. This sort of takes away variability because of year and field. There were some years where there was a lot of weeds and a lot of cover crop, and some years where there was really little because we planted a little bit late, just given when we harvested. And then on the y-axis is the white grub density. So to measure white grubs, we excavated 1 foot by 2 feet by 1 foot sort of pits, put that all out on this tarp, and then sorted through it to get the number of white grubs in the volume of soil. And we did this across the field. And what we found was that there was a negative correlation between cover and white grub populations, which is sort of what we expected, but it means that where there's higher cover biomass, either it was a cover crop planted or there were more weeds, that those white grub populations were lower. Our next question were, do insecticides and cover crops reduce damage to the plants? And we did this. We went through the field and looked at 20 individual plants across the field. And we sort of rated them from 0 to 4. So a 1, for example, would be 10% of the leaf area had been removed or less than that. 2 would be less than 25% of the area removed. 3 would be less than 50%, and 4 was sort of between 50 and 100%. And just to give you a sense of what damage looks like, so this was an example of the damage that we saw. So this would be rated as a 1. So I'm going to first start with the soy, because the story depends on what crop it is. So in soy, when we combined all years, we found that the control, so the no pest management treatment, had the highest level of damage, and it was significantly higher than the seed treatment damage. So seed treatments sort of protected the crops a little bit, but just remember that this 1 was less than 10%, so way below sort of a concerning threshold for soy at this time of its life cycle. When we look at corn, we see quite a different story. So corn, it depended on what year we were in, but in year 2017 and 2018, the cover crops actually reduced plant damage significantly. So you can see that the cover crops had less damage than the no cover crop treatments, regardless of what the insecticide treatments were. In 2019, the story is a little bit more complicated. There is an effect of insecticide. It interacts with the cover crop, so it depended sort of on what cover crop and insecticide it was. But generally, you can see from this graph that cover crop generally reduced damage. So our take-home message here is really that there was no effect of neonics or IPM in corn. It has a limited effect on soy, but it probably doesn't matter all that much unless you have really high populations of pests. Ooh, this did not turn out well. Okay, our next question was, are predators affected by insecticides? And this picture here is a little carabid beetle sticking its little nose out of a hole. But what we have we measured predator activity density using pitfall traps. So we have little carabid beetles or spiders wandering across the soil, and they come across their pitfall trap and you fall in. How a pitfall trap works, right? So what we found was, I'm going to show you total predator activity density. These total predators include ants, carabid beetles, rove beetles, harvestmen, and spiders, and a few centipedes in there too. So there's sort of any ground dwelling predator that was falling in, was mostly predaceous and falling into our pitfall traps. We counted as part of this sum. So in 2017, you can see that I'm only showing the no pest management and the preventative pest management. That's because we applied the insecticide in the IPM treatment in 2018, so it didn't make a ton of sense to show it for 2017 since it wasn't different from the controls. So I'm going to show you each individual field in each year. So in corn, we found in 2018, when we applied that IPM treatment, that it reduced the predator activity density a lot, more than the preventative pest management treatment reduced the predator activity density, but both of them sort of negatively affected predators compared to the control. In 2019, predator activity was much, much lower, probably a combination of weather and when we were sampling. Right after a rainstorm, the predators tended to hide a little bit more. So we didn't get quite as much activity, but we see sort of a rebounding of the predators in the preventative pest management treatment. In corn, we see a similar trend in 2018, where the IPM treatment reduced the predator activity the most, and we sort of have an intermediate level of predators in the preventative pest management treatment. So if we combine this with cover, we can look at the interaction between insecticides and total cover. So here is total cover on the x-axis and total predator activity density on the y-axis for corn and soy in each year. And in corn, we see a general trend up and a general positive correlation between total cover and predator activity density, which means that the more cover you have early in the spring, the more predators you're going to have in June, which makes sense. It sometimes depends on what the insecticide treatment was. And in 2019, in the preventative pest management treatment, it was actually a negative trend. This may, it's hard to interpret that exactly with 1 year and 6 plots, but overall, there was a positive relationship. We don't see that same relationship in soy. So if you'll think back to what we found for the pest damage to plants, pest damage was lower in the cover crop plots in corn, but not in soy. And this might be because those cover crop plots have more cover in the spring, they foster more predator activity and reduce pest populations and thus damage. So we then wanted to know how those predators, how good they were at eating prey. So we sort of staked out some waxworm caterpillars. These are just feeders for pets. They're pests in honeybee colonies. We stake them to a little piece of clay with a pin, and then we set them out in a cage. So this prevents things like mice from coming in and eating them. So we're trying to get an invertebrate, a small invertebrate population sort of quantification that are able to reach these and not mice. So we tend to see a lot of carabids and ants that get in here and start eating these. The ants really love these waxworms, so they'll come and just feast. And what we see here, so we're measuring predation rate across each of these plots with and without the cover crop in our no pest management and preventative pest management or IPM treatments, depending on the year. So in corn, it's kind of a hot mess. Early in the season, we sometimes see a reduction in predation rate in the preventative pest management treatment, but there is not a really clear or consistent take-home message. Same thing for soy. The predation rates didn't necessarily translate, or the predator populations and the pest populations didn't necessarily translate to changes in the predation rate. What we do see at the mid-season, so this is the end of July, really high predation rate. So almost 100% of these waxworms get eaten. And by September, we're starting to see a really interesting sort of latent effect of cover crops. So cover crops are cover crops are increasing the predation rate late in the season, even though those cover crops had been killed in May. So it's possible that some of those populations of rabbit beetles went through their larval cycle, laid their eggs, went through the larval cycle, starting to emerge and prey on the waxworms late in the season. And then finally, did it matter? Do we care? Are these insecticides improving pest management? Are we seeing sort of a problem in yields with cover crops? So we measured yield of grains in corn. It was in October, November, and soy it was a little bit earlier. Across these years, we don't see any effect of either cover crop or the insecticide for corn yield at all. Soy, we see a similar trend. There's some effect, there's some negative effect of the IPM in 1 year, a year after it was applied in soy, and there's an increase in yield because of the cover crop in soy in this 1 year. So really those insecticides are not providing a yield benefit. So they cost more money, but they're not providing a yield benefit. And really what we found was that the IPM treatments that we used were worse for predators than the preventative pest management treatment, which was not really what we were expecting to find, but it makes sense because we had to use this granular pyrethroid, which tends to be even more broad spectrum than neonicotinoids, and it hits spiders and other non-insect arthropods harder than the neonics do. And so we see those are important players in our ecosystems. We also found that early season cover increased predator activity and was actually better at protecting those plants than the insecticides were. So if we come back to our original questions, do preventative pest management treatments sort of cause problems or decrease the benefits of cover crops? Not really, but also those cover crops are performing really important roles. And the IPM treatment, are they better? No, not unless we use some other insecticide besides a granular pyrethroid. We were trying to apply this to soil pests, which is the same thing as those neonics or those seed treatments are targeting. So it's a really good comparison if you have soil pests. So we found that cover was correlated with less pest damage in corn. This then also was potentially because there were more predators because of the cover in corn. Mostly the insecticide and the IPM treatment didn't do what we wanted. It did not decrease white grub populations, and it really hurt the predator community and disrupted even yield in 1 year in soy. And finally, those cover crops are increasing predation late in the season, which I think is really interesting because it had been months since those had been planted. And the yield effect, so the cost benefits really do not favor using these insecticides. So our sort of final message is that planting cover as a first defense can be really important, and save those seed treatments for when they are needed, when you have soil pest populations that are high enough to justify justify treatment. So there's a whole host of people who have worked on this project. We had a lot of help in the field. And with that, I will take any questions, and feel free to email me if you don't think of a question right away or you want to talk about anything. All right.
Thank you so much. That was great. Do we have any follow-up questions?
Hey, Elizabeth, I have a question. So
yeah,
I might miss, but is this actually the rotation system or just continuous corn or continuous soybean?
So it was a rotation. So we did, one field had corn, soy, corn, the other one the other way. Because I analyzed them each individual year in fields separately, I just kind of showed the corn and the soy. We couldn't look at the effective field and the effective the crop at the same, like in the same analyses, because we didn't have it completely, we couldn't separate it completely. So
Okay.
it just, the data just made sense that way. So that's
Okay.
how we.
So if this is a continuous corn or a continuous soybean, the data may change a little bit?
Well, we're
But still-
definitely, yeah.
Okay. Some growers, do continuous corn or continuous soybean.
Yeah, certainly the pest complex would be different. We'd have tended to see more serious like corn pests or soy pests, I think, if we had sort of done continuous cropping. Yeah.
Okay, thanks.
Hello, this is Mark Hepner. Can you hear me?
Yes.
Hi. Is there a difference in cover crop? Like if you just put like winter wheat, or if you put some type of alfalfa clover mix or something like that?
Yes. So we ended up going with a grassy cover crop. So we did rye or oats, depending on when we could plant it. There were some years when we were harvesting in the snow, so we couldn't plant a cover crop. But certainly it's possible that our results would have changed depending on what the cover crop was and how that cover crop provided resources to predators. So something that had more floral resources than a grass might have in fact improved and the predator community even more, or changed the predator community. So yes, I think the answer to your question is yes, but.
Yep, thank you.
All right. Well, thanks to Dr. Rowan and all of our presenters for today. We did run over a little bit, but it's so infrequent that we get together that I hate when we're having good conversation to cut anyone off. And that's definitely the one thing, although virtual is at least something, I really do miss seeing everybody in person. And I'm cautiously optimistic that in 2023, we will possibly get to have an in-person meeting again. And that's something that we'll be discussing in the business meeting today. I think that if we have it in person, where will it be? Will we go back to North Bend, or will we maybe do it somewhere else? So again, I just want to say thank you. And Dr. Park was actually able to record the session today with all the presenters. So hopefully here in a little while, we'll be able to send out a link. If you know any members who weren't able to attend today but would like to watch the recording, then we will have that. And that concludes the presentations. And for those who wish, you can stick around, and we're going to talk about treasury stuff and old business and new business and all our inner workings. So appreciate